Hello everyone. Professor Frina, can you please listen me?
Hello, can you hear me now?
Good evening, Professor Frina.
Yeah, my video is not working. I don't know why. Can you hear me?
Yes, I can hear you.
Yeah, but my video is not working. I don't understand why.
Okay, let me check.
Hello, can you hear me now? Do you want me to log off and log in again?
Yes, Professor Frina.
Yes.
Professor Farina, if you are speaking, I cannot hear you.
और डॉक्टर विक्रांत साहब, हेलो। जी अस्सलाम वालेकुम प्रोफेसर नासिर साहब। सर कैसे हैं आप? ठीक हैं? होप एवरीथिंग इज ग्रेट ऑन योर साइड।
Walaikum assalam. How are you, dear Jubran?
Sir. Ji, Assalamu alaikum, respected Professor Farina Saiba. Ma'am, hopes everything is great on your side. Ma'am, you can unmute now. You are the co-host now.
Hello.
Yes, sir. You are audible.
Thank you very much.
Ladies and gentlemen, we will begin shortly.
Respected Lauren, can you please listen to my voice?
Yes, Dr. Gibran.
Okay, there was a query from Dr. Lauren that not able to listen to the voice of any meeting participant. I think no one was speaking at that time.
Dear Jibran, what is the reason we are not starting the program? Actually, we are the 8 minute late.
I think there is no solid reason. We have the participants right now all across the globe, and even we have some very good number of students from different universities. Those have joined from a collective account, and I request Dr. Sayam to start the session as soon as possible. Dear Siam, do we have anything left? I think there is nothing. You can now start the session.
Yeah, we are just going to start the meeting.
Please.
Let me give 1 more minute, please.
डॉक्टर नासिर था माइक उतार लो गर्मी बहुत ज्यादा वा
अच्छा डॉक्टर साहब लो उतार लिया सर मैंने होर भी सर मैंने बहुत सारी ना सेटिंग चेंज करनी पैनी है इसलिए नहीं उतार सकता और ठीक हो सर
अल्लाह का शुक्र है तुम सुनाओ जिब्रान साहब ठीक हो
सर बिनू भी कह रहे हो कोट लाला सर शुक्र लाना अल्हम्दुलिल्लाह अल्हम्दुलिल्लाह great ਤੁਸੀਂ ਸੁਣਾਓ ਠੀਕ ਹੋ ਸਰ ਸਭ ਠੀਕ ਹੈ ਸਰ
ਬੰਦੇ ਦਾ ਸਵੇਰ ਸਵੇਰ ਫੋਨ ਆਇਆ ਐਨਆਰਸੀ ਚੋਂ ਨੇ ਕਿਹਾ ਸੁਣਿਆ ਫੈਸਲਾਬਾਦ ਬਾਰਿਸ਼ ਹੋ ਰਹੀ ਮੈਂ ਕਿਹਾ ਅੱਗ ਬਰਸਾ ਰਿਹਾ ਸੂਰਜ ਇਸ ਚੀਜ਼ ਦੀ ਬਾਰਿਸ਼ ਹੋ ਰਹੀ
ਸਰ ਟਰੂਲੀ ਅੱਜ ਬਹੁਤ ਜ਼ਿਆਦਾ ਗਰਮੀ ਦੀ ਪ੍ਰੈਡਿਕਸ਼ਨ ਹੈ 43 44 ਦੀ ਪ੍ਰੈਡਿਕਸ਼ਨ ਹੈ ਕੱਲ ਤੋਂ ਹੋ ਸਕਦਾ ਹੈ ਕਿ ਬਾਰਿਸ਼ ਸ਼ੁਰੂ ਹੋ ਜਾਏ Assalamu alaikum, respected ma'am. Professor Farina, hopes everything is good on your side.
Professor Frina, can you please listen us? Okay. So, hello and a very warm welcome, distinguished keynote speakers, respected session chairs, faculty members, graduate researchers, industry professionals, and ladies and gentlemen, welcome to the second International Graduate Research Conference organized and hosted by the Poultry Professional Society in conjunction with the Department of Poultry Production, University of Veterinary and Animal Sciences, US, Lahore. This conference is supported and sponsored by Gulf Poultry Magazine. So, 2 years ago, basically PPS began with a simple vision to create meaningful opportunities for young researchers to engage with international experts and contribute to scientific advancements. Today, that vision has grown substantially. GRC 2026 brings together more than 20 international recognized keynote speakers and participants representing universities, research institutions, and industries across the globe. Our objective extends beyond presenting research. We seek to encourage collaboration. We seek to inspire innovation. So most importantly, we seek to empower the next generation of poultry scientists who will address future challenges in food security, sustainability, animal welfare, disease prevention, genetics, nutrition, and production systems. So once again, thank you all for joining us today. So let us begin this 2-day exciting scientific journey together. So first of all, I would like to invite our first conference chair, Dr. Gibran Hussain, Chairman, Department of Poultry Production, US, Lahore. So it's over to you, Professor Dr. Gibran. Thank you for joining us.
Thank you very much, Sayam. Just before the start of my welcome address, I'm worried about Dr. Farina Hadak, who is unmuted now, but she is unable to.
Can you hear me now?
Video.
Can you hear me now?
Yes, ma'am. Assalamu alaikum.
Yeah, walaikum assalam. I can hear you, but my video is saying that the host has not allowed me to switch on my video.
I'm sharing the same with the host that there is some problem with the video of Professor Farina. So Syam, please check it out and enable the video as well. Thank you very much.
I can speak.
And I'm feeling great honor while having you with us at this graduate research conference. Being the former member of this department, the Department of Poultry Production, and we always consider you as the present member of the Department of Poultry Production as well. And whenever we need to seek some guidance, and we do have in our mind that one of our colleagues is there in the UK and she is doing great with the science and the research in the field of poultry production. So a big thank you, respected ma'am. And overall,
Professor
Assalamu
Fina, can you
alaikum.
please check your, Dr. Gibran, just give me a few minutes.
Sure.
Professor Fina, can you please check your
Yes, done
video
now.
camera right now?
It's okay now.
Yeah, can you see me now? Yeah, I think it's working
Wonderful.
now. Fantastic. Yeah, thank you. Yeah.
So a big thank you, respected Professor
Thank you very much for a
Farina,
very warm welcome. Thank you.
and I can understand that it's about 5
That's
in the
okay.
morning, early
Yeah.
morning, and you're there with us. And honestly speaking, we feel great pleasure. So, dear colleagues from all across the globe, Assalamu alaikum, hello, and a very good morning to all of you, especially the distinguished speakers from all across the globe, then the researchers, faculty members, and students, and the industry leaders as well who have joined this forum. And I think it's a great honor for me that we are able to connect and get the words of wisdom from the colleagues who are leading the research and academia in the field of poultry production. And again, we are trying to work as a bridge between the global academic leaders and the students, prospective students who would be serving this industry, who are serving this industry with the vision, with the wisdom, and the learnings that our academic leaders and the research leaders have gained through. Here I am finding some very, very great names with us, and it would not be possible to individually name them, but I feel honored to say thank you to all of you. And I think the sector for whom we are trying to bring innovation, we are trying to bring bring new ideas, we are trying to bring sustainability, is a sector that is playing great role, vital role in ensuring the food security, nutrition of the masses all across the globe. As the industry faces emerging challenges, this specific industry is facing emerging challenges. Hence, research, innovation, and sharing of that research and innovation is extremely important. And at this particular stage, I share my sincere thanks to the members of the Poultry Professional Society, specifically Dr. Sayam Akram and Mr. Umair, his team member, and the whole Poultry Professional Society, as well as my department, Department of Poultry Production, especially Dr. Muhammad Waqas, to effectively coordinate this wonderful activity. And I really commend all the team members of the Department of Poultry Production while ensuring the availability of the speakers, globally renowned speakers, and then connecting these speakers with our industry leaders and with our students. So a big thank you to all the participants, and specifically to the distinguished speakers. I would like to share that we have connected more than 12 universities from all across Pakistan, and students from different universities are there, and they are basically sitting in their classrooms and seminar rooms, and one of their faculty member has engaged those. So if we are seeing total number of participants is 31, but many of them are representing a good cohort of the students from their respective institute. So a big thank you to the keynote speakers, distinguished speakers for their time and efforts in making this a wonderful learning session for our students. Thank you very much. Jazakallah.
Thank you very much, Dr. Gibran Hussain, for your wonderful remarks. Now, I would like to ask our second conference chair of this conference, Dr. Nasir Mukhtar, Associate Professor of Poultry Nutrition at Institute of Animal Sciences, BMS, Arid Agriculture University, Rawalpindi, Pakistan. Dr. Nasir Mukhtar also serves the WPSA Board of Directors. I would like to ask Dr. Nasir Mukhtar to please give your welcome remarks. Thank you for joining us. Dr. Nasir Mukhtar, it's over to you.
Thank you very much, Mr. Syam. This is Dr. Nasir Mukhtar, the Director of Poultry Advisory Committee, the Conference Chair of the Graduate Research Conference 2026. Assalamu alaikum and very good morning, ladies and gentlemen. It's a pleasure to welcome all distinguished speakers, researchers, students, and industry professionals in second Graduate Research Conference 2022, which is organized by the Poultry Professional Society in collaboration with the Department of Poultry Production, University of Veterinary Animal Sciences, Lahore, Pakistan. The Poultry Professional Society was established with a vision to strengthen scientific communication, professional networking, and international collaboration in the poultry science. Through initiatives such as this conference, and Poultry Professional Society aims to create the meaningful opportunity for graduate researchers to engage with leading experts, share innovative research, and build global academic partnerships. The remarkable growth from the Graduate Research Conference 2025 to the Graduate Research Conference 2026 reflect the increasing importance of international cooperation in addressing challenges related to the poultry health, nutrition, genetics, poultry welfare, sustainability, environment, and food security. I encourage all participants, particularly our young researchers across the globe, to actively engage in discussions, explore collaboration opportunities, and continue contributing to the advancement of the poultry science. I congratulate to the organizing team, particularly Dr. Syam, Mr. Waqas, Dr. Gibran, and all the speakers, and thanks all keynote speakers and participants for supporting this important scientific event. I wish you a productive and successful conference. Thank you very much.
Thank you very much, Dr. Nasir Mukhtar, for your such a wonderful welcome remarks to our audience and to our distinguished keynote speakers. So we now move to our scientific session number 1. This session will focus on poultry nutrition and alternative feed resources. I would like to invite our distinguished session chair, Professor Dr. Farina Khattak, who is the Professor of Applied Poultry Nutrition from Scotland's Royal College, United Kingdom, to provide opening remarks and guide the session proceedings. It's over to you, Professor Farina. And thank you very much for joining us.
Thank you, Simon. Hello, assalamu alaikum, and a very good morning to everyone joining us from Pakistan and from around the world. On behalf of the Poultry Professional Society, Pakistan, it's my pleasure to welcome you to session 1, which is poultry nutrition and alternative feed resources as a part, and this will be the first session as a part of the second International Graduate Research Conference 2026. The poultry industry today faces one of the greatest challenges in its history, that is producing more food with fewer resources while maintaining animal health, welfare, economic viability, and environmental sustainability. Nutrition sits at the very heart of the challenge. Today's session brings together leading scientists from 4 different continents, each offering a unique perspective on how nutrition can shape the future of poultry nutrition and poultry production. From the session will cover bone health, embryonic development, and it will continue to gut programming, alternative feed resources, and circular nutrition systems. Our speaker will explore some of the most exciting developments currently influencing poultry science globally. I would really like to encourage everyone, particularly our graduate students and early career researchers, to actively engage with the discussions and ask questions and take advantage of this opportunity to learn from internationally renowned experts. With that, let us begin our scientific journey with session 1. Our first speaker, Simon, do we have our first speaker online?
Yes, Professor
Professor
Woo Kim is
Kim.
available now.
Okay.
Yeah,
All right, okay.
I'm here.
So our first keynote speaker is Professor Woo Kim from University of Georgia. Professor Kim is internationally recognized for his work in poultry nutrition, specifically on mineral metabolism, bone health, and nutrient utilization. His research has significantly contributed to advance our understanding on how nutritional strategies can improve laying hen performance, skeletal integrity, and overall welfare. Today, Professor Kim will talk about role of vitamin D and its metabolite on bone health and in laying hens. Please join me in welcoming Professor Kim. Professor Kim, over to you, please.
Okay. Yeah, thank you very much for nice introduction. And I'd like to share my slide. So can you see my slide?
Yes, we can
Okay.
see your slides.
Yeah, that's good. Yeah, so today I'm going to talk about the role of vitamin D and its metabolites on bone health in laying hen. So, like skeleton is very important for overall animal health and also growth and development. But, like a laying hen, because they produce the egg every day. So this bone is very important because a lot of eggshell calcium comes from the bone. So often older laying hen has several bone issues, including osteoporosis. And also this is important animal welfare and also economic issue because bone defect actually affect egg production and also shell quality. So as you know, laying hen has a specialized bone compartment called medullary bone. So, this medullary bone is formed inside the bone marrow. So it's very completely distinguishable from the cortical bone. Cortical bone is very thick and then dense bone, but this medullary bone is woven bone, and also there's a lot of mineral actually accumulate, and also there's a lot of bone building cell, osteoblast, and also osteoclast there in medullary bone. So this medullary bone is very important for eggshell formation in laying hen. So this is some of the histology slide. So you can see the left side is a toluidine blue staining. So it actually clearly shows the difference between the cortical bone and medullary bone. So medullary bone is not dense. So there is a lot of space, surface area. So this is actually beneficial. Those surface area, there's a lot of osteoclast, the bone resolving cell actually exist. And then whenever this hen need the calcium for eggshell formation, this osteoclast bone breaking cell activate remove the calcium and then provide calcium for eggshell formation. So right side slide, you can see the red marked one is actually osteoclast. So activated osteoclast produce the tartrate resistant acid phosphatase. So these actually, they produce this one. So this is staining actually show the active osteoclast. So you can see that this red colored cell, surface of medullary bone is osteoclast. There is osteoclast, a lot of osteoclast on the surface of the medullary bones. This osteoclast is multinuclear cell. So they resolve this bone, and then quickly resolve bone provide the calcium for eggshell formation. So generally, like 60% of eggshell calcium comes from the diet, and approximately 40% of eggshell calcium comes from medullary bone. So large, huge amount of calcium actually resolve the this medullary bone every day. So like this graph actually showed as laying an age, so total bone volume increase, but in total bone volume, specifically medullary bone volume increase, and at the expense of structural bone. So here you can see the cancellous bone, one of the structural bone, actually reduced as they age. So medullary bone volume continuously increase, and then during the this increasement, there's the osteoclast that continuously resolve the cortical bone and cancellous bone and increase the medullary bone. So that actually make the structural bone more thinner, and also later on in the older laying end is a thin cortical bone or cancellous bone actually reduce the skeletal integrity, and also there is a lot of incident for bone breakage. So like that's actually important welfare issue, because there is a lot of pain involved. Also, this bone issue affect the eggshell quality and also egg production. So it is important economy issue as well. So this is another data. We use the micro CT, and then so we actually measure the bone mineral content in cortical bone, medullary bone, and trabecular bone. So micro CT can actually separate, scan this like a bone, and then create 3D imaging, and then we can separate different part of bone and then evaluate separately. So you can see here, once they lay egg, start lay egg, and you can see the green line, cortical bone continuously increase, but like the week 18 or 19, once they start laying egg, this cortical bone mineral content is a continuous decrease. But like a medullary bone, their mineral content continues to increase, and also the trabecular bone, also the slightly there's a trend to reduce the as they age. So these initial egg laying is important like a stage, because once they laying egg, then cortical bone continuously reduced, and it is very difficult to increase the mineral content, mineral density in cortical bone. So, it is important to improve the bone quality, bone mineralization before onset of egg production is very important. So normally, like egg production and egg quality, always we think about during laying period is important, but it is important. But to do that, also the before laying, the rearing period also very important. So you can see that this picture, top one is a healthy normal hand bone. So there's a thicker cortical bone and thicker cancellous bone. There is a connect each other very tightly. But bottom picture is osteoporotic laying end bone. You can see the outside cortical bone is thinner, and also cancellous bone also thinner. There is a connectivity is loosened. So like they are very easy to break. So why the osteoporosis happen in laying hen? So, one first reason is genetic selection for small body weight, early egg production, and high egg production. So, and second reason is suboptimal bone growth in early egg production. So like I mentioned, once they start laying egg, then it is difficult to improve the bone growth and bone mineral accumulation. So, like we want to laying hen produce the egg earlier and then longer, then it's actually affect the farm economics. But laying hen health perspective, it is not good practice. And also the limited activity. So, like normally still a lot of like a farmer actually use the like a caging system. So, like cage, we put several bird in small cage. So their activity is limited. Then also it affect bone development and bone health. So, but also nowadays there is trend in poultry laying industry, like changed from the caging system to the cage-free or aviary system. So, so give them more space to move, actually improve the bone health. But like also like a cage-free system or aviary system also cause another like increased another bone issue is kill bone damage. So they can fly and then like a bump into object, and then there's a kill. Kill is damaged, also breaking, also another issue. So how we can improve the skeleton integrity and prevent bone disorder in laying? So first we can think about is balanced and adequate calcium and phosphorus supplementation. So calcium and phosphorus is are most important mineral for eggshell formation and also bone health. So that's like a fundamental importance. But also we need to think about is how to stimulate like a bone growth development before onset of egg production in laying hen. So yeah, I'm gonna more talk about how we can stimulate bone growth development before onset of egg production. So if you look at this is like a bone mineralization. So we always think about calcium and phosphorus important for mineralization. It's important, but we need to understand about like there's two steps of bone mineralization. First step is deposition of osteoid, which is like a bone matrix. So osteoblast is actually differentiated from the mesenchymal stem cell. This osteoblast is a bone building cell. So they first produce the bone matrix protein, and then second step in mineralization. Under the newly formed bone matrix protein, actually attract the calcium and phosphorus, and then make the mineralized bone. So without this bone matrix protein, like calcium and phosphorus accumulation on the bone is very limited. So these osteoblasts and also producing bone matrix protein is important for bone mineralization. So can vitamin D and its metabolite stimulate bone growth rate development before onset of egg production? My answer is yes. So whenever we think about vitamin D, like we always think about the intestines. So vitamin D can increase the calcium and phosphorus absorption, and then those can help the bone mineralization and bone formation. So that's absolutely right. But vitamin D has a dual function. So not only in the intestine, but also they can affect directly bone and then stimulate like a mesenchymal stem cell differentiate into more bone cell, then this bone cell produce the bone matrix protein, which attract the mineral and increase the mineral deposition, and it actually helping like a bone formation and bone development. So so we always think about vitamin D, not only the intestine for mineral absorption, also important for directly influence the bone mineralization, bone matrix formation in the bone. So our lab actually isolate chicken mesenchymal stem cell, and then we can differentiate into the osteoblast bone forming cell, and then in the cell culture we can actually measure measure the which like a bioactive component actually stimulate bone mineralization. So this is actually very useful tool. So like we have a potential feed additive or some of the bioactive molecule, then using this mesenchymal stem cell system, and then we can check which one has a pro bone effect, and then using that one as a feed additive to stimulate bone development in animal. So this is like a mesenchymal stem cell culture system. So we use the different dose of 125 the vitamin D. So vitamin D. So you can see the Algerian red and Boncasa staining. This actually show the staining, show the mineralization. So vitamin B12, there's a dose as it vitamin 125 vitamin D3 like increased, and there's a mineralization also increased. More darker red is more mineral accumulation, and Boncasa staining also the like a higher 125 vitamin D3 actually increased the bone mineralization. So like this actually prove vitamin D not only increase the bone calcium absorption in the intestine, but directly influence the bone cell formation and increase the mineralization. So like for laying and diet and poultry diet, we always add vitamin D3. So vitamin D3, as you know, it is not really like an active form, but it should be converted into 25 hydroxy vitamin D in the liver, and further converted into 125 vitamin D3 in the kidney. And then did actually increase the calcium and phosphorus absorption and also bone mineralization. So, like these like metabolite isoform, so 25 hydroxyvitamin D, even 125 125 dihydroxyvitamin D is more like a bioactive than vitamin D3, and also sometimes older hen has a liver issue or kidney issue, then it actually limit the conversion of vitamin D3 into the active form. So this isoform is more active, and then it's commercially available, so we can actually evaluate this one for the bone mineralization in laying hen. So this study actually evaluate effect of dietary 25 vitamin D on bone development in pullet and also laying period. So hypothesis is increase the bone growth by this vitamin D before onset of laying, actually maintain bone health during laying period. So we use the Highline W36 pullet, like a bring in, and then pre-treatment. So first treatment is just the normal vitamin D, and second treatment is double dose vitamin D. And then third treatment was normal vitamin D with 25 hydroxyvitamin D. So we actually conducted this study until week 95. So, but today I'm gonna show some data until week 60. So week 18 was onset of laying. So bone evaluation, we measure the bone growth rate using calcium injection, and also using DEXA X-ray machine for whole body bone mineral density, bone mineral content, and using micro CT, we actually did structural analysis. So bone growth rate using calcium injection. Calcium is a bone-seeking dye. So at week 10, we inject the calcium for first injection, and then 10 days later, second injection. After 4 days after second injection, we sacrifice bird and then collect bone and then make the bone slide. And then we actually under the fluorescent microscope, we can take a picture. So you can see there are two calcium lines. So the inner line is first injection, and then second line is like a second injection. So during like a 10-day interval, how much bone is growth. So distance between these two calcium label, we can measure the bone growth rate. And also using DEXA, we use the whole body mineralization and bone density. And also using micro CT, we use the we did some of the structural analysis and all bone mineral density and bone mineral content. So we actually selected this part of bone because this part of bone contained the cortical bone, cancellous bone, and also medullary bone. So we can actually separate these three parts and then evaluate also separately. So this is the actual micro CT image of laying and bone. So using this micro CT, we can scanning, create a 3D image, and then we can separate cortical bone, cancellous bone, and medullary bone, and then evaluate separately. So it is a very powerful tool. So using this micro CT, we can measure bone volume, bone mineral density, and also porosity, how much hole actually in the bone, and also trabecular bone structural analysis. So result, we measure the serum 25 hydroxyvitamin D level. This is like a standard measurement for vitamin D status. So as we expect, gray line 25 hydroxyvitamin D treatment show the highest serum 25 hydroxyvitamin D3, and then double dose show the middle, the orange line, and then single dose vitamin D show the lowest serum 25 hydroxyvitamin D level. And during the rearing period, 0 to 17 weeks, we measure the bone growth and development. So this is a micro CT image. So this is bone mineral content. So interestingly, we actually expect the 25 hydroxyvitamin D actually increase the bone mineral content, bone mineral density, but bone mineral content, there is no significant difference. There is no change. And then we measure bone mineral density. Bone mineral density is one of the key indicators for bone health. But interestingly, this 25 hydroxyvitamin D treatment significantly lower bone mineral density. This is a completely opposite result we expect. We expect the 25 hydroxyvitamin D improve bone health, so increase the bone mineral density. So it was a surprise result, but like if we use some conventional assay, then we might conclude 25 hydroxyvitamin D actually reduce the bone quality. But this microCT is a powerful tool, so we actually further evaluate bone volume, structural analysis, and figure out why this vitamin 25 hydroxyvitamin D reduce the bone mineral density. So we measured also bone volume. So you can see here 25 hydroxyvitamin D actually increase the bone volume. Bone size is bigger. But before I showed the bone mineral content there was no difference. So basically, the reason reduced bone mineral density is bone mineral content is same, but bone volume is increased, then it reduced the bone mineral density. So we also measured porosity. There is pore volume in cortical bone. Also, 25 hydroxyvitamin D increased the pore volume. So it's a big vitamin 25 hydroxyvitamin D make the bigger bone, and then there is a lot of space inside the cortical bone. That's why it reduced the bone mineral density, and also the total bone tissue volume also increased by 25 hydroxyvitamin D, and also 25 hydroxyvitamin D increased the trabecular bone volume, and also even bone marrow volume is increased. So basically, 25 hydroxyvitamin D during a rearing period, it actually stimulate bone growth and increase the bone volume, and then there is a lot of space inside the cortical bone. So we don't know whether this change by 25 hydroxyvitamin D is beneficial for bone health during laying in or not. So let's look at whether this one is beneficial or not during the laying period. So bone quality and 60-week, bone volume still significantly higher by 25 hydroxyvitamin D. But also bone mineral content also significantly increased. So which means during rearing period there is a lot of hole is actually filled by calcium and phosphorus. So 25 hydroxyvitamin D increased the bone mineral content. And also bone mineral density. Bone mineral density is actually same level. It's numerically higher, but like a rearing period, 25 hydroxyvitamin D significantly reduce the bone density because there's a lot of space inside the cortical bone. So this data actually show the 25 hydroxyvitamin D supplementation during laying period actually filled all the space and then make the bone more mineralized and bone more strong, healthier bone. And also the inside of trabecular bone connectivity is 25 hydroxyvitamin D significantly increase the connectivity. You can see the green. And also the DEXA whole body like a bone mineral density also significantly increased by 25 hydroxyvitamin D, and also whole body mineral content also significantly increased by 25 hydroxyvitamin D. So in conclusion, this 25 hydroxyvitamin D during rearing period, it stimulates bone growth and then increase the bone size, but there's a lot of space. But during the laying period, this space is filled with calcium and phosphorus and increase the bone quality. So, like a take-home message here is good start is important. So we need to maximize the bone frame and mass before onset of egg laying. Then that really helping like a bone health during the laying period. Okay. Yeah, thank you very much for your attention.
Thank you very much, Professor Kim, for an excellent presentation and for highlighting the critical role of vitamin D in supporting skeletal health and productivity in laying hen. I'll kickstart the question answer session with the question. In commercial laying hen, because you have an extensive experience in that, my question is, vitamin D is normally added in laying hen diets in a premix. Based on your experience, do you think that is enough for laying hens during whether it is rearing phase or laying phase, or do you think additional vitamin D is required to maintain a good balance?
I think the industry continuously improve the laying hen, so increase the egg production and shell quality. So definitely there is a high, pressure on the bone especially in laying hen. So I think that we should increase the vitamin D level, especially using 25 hydroxy vitamin D. So, a 25 hydroxy vitamin D is expensive. So I think we might mix with regular vitamin D with 25 hydroxy vitamin D. So then, there's a additional benefit by the 25 hydroxy vitamin D, we can have. So also the 25 hydroxy vitamin D is improve the bone growth, but also once we increase the vitamin D level, then also we need to revisit calcium and phosphorus dietary level. Also, we should revisit there.
Okay, thank you very much. We have plenty of questions, but we will have only time for 2 quick questions. We can start with Regina. Can you switch on your mic and ask a question? Because I don't see your question in the chat box. So please unmute your mic and ask a question.
Professor Prina, we have to continue the question from the chat box. Participants are not allowed to unmute themselves.
Oh, I see. Okay, but I can't see. Okay. I can't see any question here. Okay, so I'm typing. Okay, Regina is typing, so we will wait for. While Regina is typing, Professor Kim, Simon, do you have a question for Professor Kim, or otherwise I can shoot another question?
No, I do not have any question at this moment. But I do want to mention that Professor Kim has an excellent expertise in poultry nutrition, and we are very much honored that he was the part of our first international graduate research conference as well. So let me mention a few points about him. That Professor Kim is also awarded with the prestigious awards from Poultry Science Association Poultry Nutrition Award, and also the D.W. Brooks Award, Faculty Award for Excellence in International Agriculture and Environmental Sciences. So again, we are very much thankful to Professor Kim for joining us at this time as well.
Yeah, and actually, it's an honor to come again and present my research. So I really appreciate inviting me as a keynote speaker again. Yeah, thank you very much. Yeah.
Thank you,
Just wait for another, just 1 more minute. Because we are, I put it on the timer and we are running out of 30-minute time. So Regina, so Regina is asking, so what age is 25-OH supplemented, and during supplementation, during lay with more calcium ingredient, does the addition change the metabolism?
Yeah, I think that's a really good question. So I think here I just briefly show the treatment here. So this 25 hydroxy vitamin D is 69 micrograms. So it is equivalent to the some regular vitamin D, but it has a high activity. So, like if you add this 25x vitamin D, we can see the rearing period and also the laying period. We use the same level. But we see the rearing period, definitely 25x vitamin D stimulate bone growth, but there's a lot of hole, so less mineralization per given space. So I think if you add this 25x vitamin D, we need also increase the calcium and phosphorus level. Then probably during rearing period, we can add also the limit bone mineral density reduction by this additional calcium and phosphorus. But still, I think the calcium and phosphorus ratio should be maintained during the rearing period as well. Yeah.
Thank you very much. I think we are running out of time. So thank you very much, Professor Kim, once again for a wonderful presentation. And we are really grateful for your time and your presentation. So thank you very much. We have more questions, but I hope you can look at the chat and answer the participants there, because we are really running out of time. So thank you once again.
All right. Yeah. Thank you very much. Yeah.
Just to add, Professor Dr. Kim, thank you for your excellent presentation. As a token of your appreciation, we would be delighted to recognize your contribution during the closing session, and a certificate of presentation will be awarded to you. If your schedule permits, we would be honored if you could remain with us until the end of this session.
Yeah, thank you.
Okay, we now move to
You are muted, Saim. If you are saying something, you are muted.
okay, I'm so sorry.
Yeah, we can hear you now.
So we now move to our second presentation with Professor Dr. Freena Khattak. She will present on feeding the future, circular nutrition, and alternative feed resources for sustainable poultry production. So before Professor Freena will begin, let me give a brief introduction of Professor Freena Khattak. Professor Dr. Freena Khattak is a professor of applied poultry nutrition at Scotland's Royal College, UK, contributing to agriculture and land-based engineering, monogastric science research, and related interdisciplinary centers. She is a poultry nutritionist with extensive experience in designing, executing, and analyzing commercial and academic nutritional research projects in collaboration with national and international poultry industries. Her research primarily focuses on feed resource evaluation, nutritional strategies to enhance production performance, gut health, carcass quality, and welfare indicators in poultry species. She also works on the development and assessment of feed additives to improve nutrient utilization and reduce environmental nutrient losses. In addition, her studies utilize chicken infection models to explore host pathogen interactions in disease such as Campylobacter, coccidiosis, and necrotic enteritis. Her work integrates applied and mechanistic approaches to improve poultry health, productivity, and microbial balance. So now it's over to you, Professor Dr. Freena Khattak, and thank you very much for your time.
Thank you. Can you see my slides? Because I have two screens, so I'm not sure if you can see properly.
Yes, we can see your slides.
Okay, thank you. So today, thank you very much, Saim, for a very nice introduction. So today I will talk about some aspects of circular nutrition. However, when we talk about alternative feed resources, the first question comes in mind is, why does it matter? It matters because of the global population and poultry demand projections, which indicates that by 2050, the human population will increase by 22%, whereas the global poultry demand will increase by 60%. So we can clearly see that the demand for the poultry will be significantly increased by 2050. Therefore, unless and until we work on the alternative feed resources, the resources will be so scarce that we will not be able to feed the poultry, and hence there won't be enough protein source available for human population. The next important part is, are we running out of feed? Because when we talk about feed processes, feed challenges, this is the first question that come in our mind, because everybody is worried about feed resources. But for us, as a poultry nutritionist, the most important part or question is whether we are utilizing all the nutrients that we have, or are we wasting the nutrients that we already have produced and processed and are easily available to us. The future may not be food security or feed security, scarcity, but it might be nutrient inefficiency. So the feed paradox, resource paradox, is that on one side, we have the poultry which produces, we have a high pressure on feed because of the high feed prices. The high feed prices are because there is a lot of scarcity of resources, because there is a competition for land and resources, and also the dependence on important feed ingredients. Whereas on the other side, we routinely see that a lot of valuable nutrients are lost through food waste, and some underutilized, and some of the resources are underutilized. The challenge is not simply to produce more feed, it is to recover and utilize resources more efficiently. A lot has been done on alternative feed resources. We do know that there is a lot of research done, and we have a wide range of resources which are already available, such as there is a lot of research done. If we categorize the alternative feed resources, we can simply categorize them into 5 categories. The first is protein crops, such as faba bean, peas, and lupins, which can reduce reliance on imported soybean meal. The second is agriculture co-products from the food industry, from the agriculture co and byproducts, such as rapeseed meal, DDGS, and brewer grains. The third category is from the formal food products, such as the byproduct from food industry, which is, as you can see in the photograph, breads, cereals, crisps, all these kind of products. The fifth category is marine resource, such as macro and micro algae. And the fifth category is the novel protein sources, such as insect, microbial proteins, and fermentation derived ingredients. So what are we doing with these resources? So the topic that I'm going to cover in my presentation today is on circular nutrition. And circular nutrition means that are we using better use of available resources and keeping nutrient within the food system? So today I will talk about 3 approaches. The first is the circular nutrition through formal food products. The second is alternative marine resources through seaweed use. And third is precision nutrition through fermentation. So the main message that I want to convey at the end of my presentation is that the purpose of alternative feed resources is not to replace conventional ingredients completely or entirely. The main purpose is to diversify the feed basket so that the poultry nutritionist can then use the appropriate nutrients. Secondly, they can use the nutrients so they're not wasted. And thirdly, that they can use the nutrients which are functional units, functional feed, which can serve as a functional feed and improve the sustainability and resilience in birds. So the first example that I'm going to share with you is about the former food products, which are, these are the food industry byproducts such as bread, biscuits, cereals, pasta, etc. These products are basically or originally produced for human consumption, which are no longer suitable for retail sale due to several reasons. They could be because they are produced off spec. They could be they have passed the sell-by date, or maybe there was a manufacturing disorder, and because of the packaging not done properly, they couldn't go into the retail sector. However, that does not mean that if they have passed the sell-by date or they are not fit for selling in the retail market, they are not fit or they have lost the nutrients. The nutrients are still there, so we have to retain this nutritional value, and that can be used in poultry feed through circular economy. What do I mean by circular economy? Circular economy is where you can use there are 3-4 steps involved in circular economy. The first step is that the food that is not used in the retail market, the surplus food ingredients or the off specs which are not sold, they can be recovered and they can be reused to form the feed or converted into feed. This can then be transformed into, can be given to the poultry birds, and then the poultry birds can transform them into a very high nutritional products such as egg and meat. And this can then go to the fourth stage, which is completion of the food chain, which is the food for human beings. And hence the whole cycle is complete. And therefore we reduce the waste, we recover the nutrients, and hence we close the food and feed gap. And then that means that from surplus, we have moved from surplus to sustainability. We have recovered, reused, restored, and that is the way how I see that we can feed the future. Nutritional value of former feed products and poultry diets is, in this slide, you will see a very full table. This table is the outcome from one of the study that we conducted together with Queen's University Belfast, where we did the road mapping exercise. And what we did there was that we tried to identify and catalog UK former food products that can be that are available to the poultry industry to be used in the poultry diets. I don't want you to go deep into this table, but what I want to see is want you to notice through via this table is that there are plenty of resources which are available. But the 3 main points that I would like to highlight via this table is that there are these former food products are nutritionally rich. They are high in, not very high in protein, but they are a very rich source of energy. But the biggest problem that we realized through our road mapping exercise was that the nutritional composition vary a lot between these products. And the third was that this is used in the poultry diets. However, the inclusion level will highly change depending on the batch size, depending on the composition, and depending on which ingredient have you picked up to use in your formulation. So the next part of that project was to look into the public domain and see whether these FFPs have been used in poultry diets. And what we found was that they are being used in poultry such as layers and broilers and quails and cockerels. And the inclusion levels were the most successful one and often reported was up to 40%. The other one was the possible ones were up to 75%, but there were only few ingredients which could be used up to 75%. There was also a couple of studies which showed that the bread meal waste can be used up to 100% in the poultry diet. The next opportunity or the next ingredient that I will discuss briefly is the seaweed opportunity. This is not a conventional feed resource, but this is gaining too much popularity because marine biomass has no competition with arable land, fresh water, and other resources required for terrestrial agriculture. Seaweed is also gaining popularity because it is rich in protein, minerals, and bioactives, and it's also a very good source of omega-3. In addition to that, it can be used as a functional feed ingredient as it has a potential to support gut health and improve the resilience and resistance in birds. This was outcome from our one of our study where we did the extensive literature search to find out whether seaweed is being used in poultry diet, and we did find that there was possibility, but our focus was mainly on dulce, which you see in this photograph, which is a red seaweed. So what did our study tell us? When we tried to work on dulce, which is the red seaweed, as I mentioned, we found that on paper, this is very promising because the protein values were very good, energy values were very good. But when we look at the composition, we collected 10 different samples from different coastal lines of Scotland, and we realized that each batch extensively varied with season, with origin, and the way they were processed. Our study also showed that this variability, this differences in the composition means that you cannot take just the book value and use it for the seaweed, because each seaweed which is produced has its own nutritional composition. Sorry. So when we selected the seaweed, because we wanted to do an animal in vivo study, so in this photograph, you can easily see in the hands was the dry seaweed from the seaweed which we used in our animal trial or poultry trial. And we wanted to replace it for wheat. So if we look at the dulce or red seaweed, we can easily see that its nutritional composition is like traffic lights. The protein content is very good. Energy is amber, but the main limiting factor was salt and ash content because it was very high in chloride content and also a little bit in sodium content and potassium content. So what we figure out from our analysis was that although the seaweed, this dulce or Palmyra parmada, which was the specific species that we used in red seaweed, that was it's nutritionally viable, but salt defines the inclusion limit in the poultry diets. Based on this compositional analysis, we then developed a trial where we used 5 treatments using the inclusion level from 0% to 6%. Each treatment was replicated 8 times with 10 birds per replicate, and the dulce was only included in the finisher phase. At day 35, we analyzed the growth performance. We did some digestibility analysis. We also collected the carcass yield data and also analyzed the meat yield for omega-3 analysis. So what did we find out from our analysis? So what we learned was that different levels had different importance. If we would be interested for future to use dulce for carcass yield, 1.5% was our optimum level. If we want to use dulce for enrichment of meat for DHA and DPA levels, then 3% was optimum. But we also found out that when we increased the level above 4.5%, the digestibility started to decrease, and there was no added benefit of further increase in the inclusion level. So in conclusion, we can say that dulce is a viable partial wheat replacer for finisher broilers. However, the inclusion level are to be based on the seaweed or dulce composition, and the benefits were mainly because of the polyunsaturated fatty acid, mineral and bioactive compounds which are present, which were present in this seaweed or dulce. So that was where we tried to make full use of the nutrients which are not currently fully used. Now we come to the third approach. So the third approach was that time has changed, things have changed. Traditionally, what we were doing was that we were feeding birds just to fulfill their requirement, maintenance requirement and nutritional requirement, and as a result, we would see the performance, and that's it, full stop. Now things have changed, and traditional nutrition is history. Now what we are doing is we are feeding the microbiome. We are feeding the trillions of microbes which are present in the gut, and hence the nutritional challenge is to feed the microbiome in such a way that we can improve the gut function, and then as a result improve the performance, and in addition to performance, we produce the food which is safe for human. So the strategy is totally changed, and now the target is, a nutritional target is microbiome, not simply the feed anymore. So with that in mind, one of the solution can be fermentation strategy, because fermentation strategy does allow to transform the conventional ingredients into functional nutritional ingredients. We did a study where we used rapeseed meal, seaweed, and soybean meal, and either alone or in combination, and then we treated them with lactic acid bacteria and certain enzyme to change these ingredients into complementary functional ingredients which can be used to increase, not only improve the performance, but also to improve the gut microbiome. In addition to that, we also think or hypothesized that the fermentation work in 3 different ways. The first is that it impact the nutrient, and it increases the breakdown of complex nutrient and thus make it more available to the bird. The second hypothesis is that it affect because of the microbial fermentation, which increases the volatile fatty acid and decrease the gut pH and hence support the gut health and integrity. The third hypothesis is that it also decoy the binding and microbial competition of pathogen and hence reduce the pathogen colonization. With this hypothesis in mind, we did a study where we did a 2x4 factorial design where we used the 2 factors were challenged or unchallenged birds. The birds where the challenged birds were challenged with Campylobacter, and there were 4 treatments. One was control, one was EP100, which was lacto-fermented rapeseed meal alone. The other was EP199, which was lacto-fermented rapeseed meal plus seaweed. And the third was a protein blend, which was EP219, which was a blend of rapeseed meal, seaweed plus soybean meal. So it was a blend of all 3 fermented products. 480 male broilers were used, 10 birds per pen, and there were 6 replicates. And the growth outcome was to look at the growth performance, feed intake, volatile fatty acid, and also to look at the Campylobacter jejuni counts enumerations. So what did we find out in this experiment was that the birds, the performance was improved by EP219 only. As you can see, different colors here indicate different treatments. The red color indicates the control, whereas green is EP219. The brown or red color is EP199, and the blue color is EP100. So it is very clear that EP219, which was a protein blend of soybean meal, rapeseed meal, and seaweed, gave the maximum performance, as the FCR was optimum in case of EP219. There was not much difference in case of the grower and finisher phase. However, the starter phase difference was then also translated and reflected in the overall period, which shows that EP219, as indicated by the green bar, during day 0 to 35 overall period was significantly higher or better as indicated by the lower FCR values in the study. We also found out that butyric acid was significantly higher. VFA values showed significant differences, but the differences were mainly due to butyric acid, which was significantly higher in birds fed EP219, which is a mixed protein blend. And similarly, in case of Campylobacter jejuni colonization, we saw that EP219 was successful in reducing 0.71 log reduction in the Campylobacter colonization in birds. So we found that it not only improved the fermented feed, not only improved the performance, but it also helped us to improve the food safety. With this in mind, despite all the positive points, we found out through our different studies conducted at SRUC that we are still facing a lot of challenges in terms of relatively new ways of using alternative feed resources. And what we found was that there was quite a big knowledge gap in terms of that there is not enough supply chain database, because we did find that the differences were mainly batch to batch differences. Therefore, there should be a regular intake or update of the supply chain databases, which was very limited. We also found out that there was a gap in the nutrient composition and digestibility data sets. There was a lot of data available in terms of growth performance and alternative nutrients, but there was very limited information available on the digestibility data sets, especially in terms of available amino acids and fatty acid composition. Then we also found out that there is a need to develop rapid quality assessment tools such as specific NIR calibration, which can be quick rather than analytical composition based analysis. Another thing which was very apparent was that there is a need for conducting long-term poultry validation trial. By long-term, I mean longitudinal studies, because we have done in layers, maybe we have looked at the effect on week 20 to 24 or week 20 to 34, but we don't have a study which goes from day 0 to week 60 or week 80 to look at the whole longitudinal length of the production cycle of the laying studies. In terms of broilers, we don't have the study results from consecutive 3 or 4 trials to show the consistent results. And also we did find that based on the seaweed and fermentation data, that we do need to work on the processing method, because that is a very big gap in the market. Therefore, to conclude, we can easily say that if we want to bring everything together, then the future of poultry nutrition lies on circular nutrition. Because we did find that there are plenty of nutrients which are already available and they exist within our food system, but unfortunately, we don't have enough information to use them in food feed formulation. The second part that our studies highlighted was that there is now an urgent need to work on alternative feed resources such as FFPs or marine biomass can reduce dependence on conventional feed crops. The third most important thing is that the basic traditional nutrition is the history. Precision nutrition and gut health is something that we need to focus on, which means that we do not need to simply feed birds, but we need to manage the gut ecosystem that derive health, efficiency, and food safety. With this, thank you very much for listening, and I would like to acknowledge all the companies who supported our research. So thank you very much, and if anyone has any question, please feel free to ask.
Thank you very much, Professor Dr. Farina Khattak, for such a wonderful presentation. We greatly appreciate your contribution to the GRC 2026. And now, if anybody have a question, please share in the chat box from Professor Dr. Frina Khattak's presentation.
You can see the chat, so Simon, if there is any question, please ask.
If anybody from the dignitaries ask the questions from Professor Frina presentations, they can unmute themselves, please.
Well, this means two things. Either my presentation was so rubbish that nobody understood anything, or it was so good that there is no question.
Frankly speaking, the presentation was fantastic, wonderful.
Thank you.
There is a question in the chat box. Yes, is there any feed mill using any of these alternative feed sources in regular basis? From
Yes.
Hussein Ahmed.
Yeah, Hussein Ahmed, yeah, thank you very much for a very interesting question. Yes, in UK, the farmer food products are regularly used in almost all feed mills in very small quantities as a, I would say, replacement of some of the conventional ingredients, not 100% replacement, but yes, they are used. In terms of seaweed, there is a brown seaweed is used, but seaweeds are mainly used as a bioactive compounds. So they are used as a feed additive, as a kind of feed additive rather than ingredient itself. Whereas fermentation products are continuously used as functional feed products, again, as a replacement of partly, if it is meal, then it is used partly as a replacement of soybean meal. If it is any other ingredient, then they are used as a replacement of conventional ingredients such as corn and maize. So yes, they are used continuously in UK.
I hope, Sen, you got the answer. So now, without further ado,
so
I
we
think
can go
we should.
with the next speaker. Okay.
Yes.
So do we have Professor
Professor Eugenie
Rora?
is on
Yeah,
board.
sorry if I'm not pronouncing your name
His
right.
name is Spanish name.
Okay.
He is on board.
Okay,
Yes,
thank you very much. Yeah, sorry if I have pronounced your name not right, but thank you very much, Professor Rora. Professor Rora is from the University of Queensland, Australia. And we are also very privileged to know that he is also the president of WBSA Australia branch. So Professor Aurora is internationally renowned for his work on nutrient sensing, feed intake regulation, precision nutrition, animal feeding behavior. His research has helped bridge fundamental nutritional science with practical solutions from modern livestock production. Today, he will talk about nurturing embryonic development, a critical frontier in transition to antibiotic-free broiler system. Please join me in welcoming Professor Rora. Welcome, Dr. Rora, and it's over to you, please. If you can switch on your camera so that the audience can see you.
Yes, thank you so much.
Yeah, thank you so much. Thank you. Yes, we can see you. That's the good start.
Yes, and I'll just put it in presentation mode, hopefully.
Yeah. Fantastic. We can see your presentation too. That's very good.
Excellent. So, well, thanks so much. I'm not going to try even to pronounce it correctly, Professor Khattak. So,
yeah.
Welcome to the world of difficulties of
exactly.
pronouncing culturally difficult names for each other. So it's a pleasure. Thanks so much for the invitation, Mohammed Sayyam Akram. I'm probably also killing phonetically your name. Apologies for that. It's a pleasure to be here, and it's an honor. And I've seen a number of names in the audience, students that I'm familiar with. So it's fantastic. It's a great opportunity. Congratulations for organizing such a nice event. and you've been so successful in getting a number of excellent speakers, such as the two speakers who preceded me. So that leaves me an important job to try to keep up the level. I'm not sure I'm going to be successful. It is a respectful way to start a presentation in my country, in Australia, to first acknowledge the traditional owners of the lands where I live and I work. In my case, the area of Brisbane City and the Lockyer Valley regions are the Turrbal and Jagera tribes, and I would like to show my respect and to recognize their valuable contribution to our country, Australia, and the global society. So, as Professor Katak mentioned, I'm going to be talking about a bit philosophical, to be honest. I don't know what I had eaten when I was putting together the presentation, but it came up a bit philosophical. I want to convince everyone in this group that there is a serious gap of knowledge in embryonic development in chickens. You would think that in poultry science we know a bit of everything, if not a lot of everything. But I'm going to try to convince you that we don't know much about how to nourish the embryo. And this is a crucial aspect, particularly in this, Professor Kata was talking about circular economy, and we all want to contribute to sustainability of poultry production, reduce the use of medicines, antibiotics, etc., to avoid antimicrobial resistance. But if we want to get there, and that's an aspirational aspect that we all need to aim at, but I think it is crucial to better understand the embryonic development first, and I think we are quite a long way from there. And I'll explain to you why I think we are quite far away from this. And as you probably all know, and I hope you would agree, that if there is a weak point in the chain of a chicken life, that's the immediate post-hatch period. That's when the chicken is most vulnerable. That's when you will see relatively high mortality rates, 1%, 2%, even higher in some cases. That's when we need antibiotics. That's when we actually see negative impact on growth that lasts all the life of the chicken. So that post-hatching period is fundamental if we want to have a successful, healthy, and with excellent welfare production of chickens. Now, why are they so vulnerable? There's obviously a number of aspects, and I'm not going to deny that, but if I'm going to mention at least two of the most important, is because the embryonic development does not quite get to a maturity stage enough to have a fully functional gastrointestinal tract and immune systems. So it is a target of improving embryonic development by improving particularly these two systems, the digestive and the immune system, as a target before even aiming at bigger and better places. So the embryonic development takes place in 3 weeks. That's it, 21 days. Well, that's it, or that's a lot. If you put it in perspective of the whole life of a chicken, 21 days is 1/3 of the life of a chicken. So for being such a, if you allow me to say, forgotten a period of the life of the chicken, those 21 days is quite a long period that we have forgotten about, right? We've done a lot of research in having fantastic balanced diets. We know exactly the nutrient requirements for every day of the life post-hatch. But do we know the nutrient requirements of the embryo? And I'm not going to ask the question loudly because I know the answer. No, we don't know. We don't know the nutrient requirements of the embryo. So that's what is actually keeping me entertained. We talk about transgenerational nutrition, and transgenerational nutrition is no other thing than the influence of the prior generations, the parental generations, and how the nutrition of broiler breeders in this case can impact, can improve the development of the next generation, the progeny, right? So we do quite know very well that how to nourish, how to feed the broiler breeders. and we do that very well. But keep in mind that what we feed the broiler breeders will have an impact on egg mass, egg composition, and also therefore the embryonic development. There's also a second window, and I'm going to talk about this second window as well. So the first window being broiler breeder nutrition and how can we impact the development of the embryo through broiler breeder nutrition. But there's a second window, which is in ovo. So while this embryo is developing, can we supplement that development by injecting additional nutrients and other functional ingredients? And I'm going to be talking about this in the time that I have. Now, there's obviously other strategies that I don't have time, and I'm not going to be talking about, such early feeding strategies and so on, to improve the robustness of this chicken embryo. Now, this is a very unpleasant table, very big and busy, and I'm not, of course, going to read it all or expecting that you will read it all. Please don't. But my point is, when we think about the fertile egg, we tend to believe, and it is true for myself, we tend to believe that every nutrient that's needed for developing life, for developing that embryo, is already in the egg, right? I think you would agree with that. Well, not exactly. That's the point of this table. And this table is a literature review that goes through every deficiency that has been identified during embryonic development, even including mortality, causing mortality. And those references can go all the way from, I don't know, 1950, Sandy et al., to very recent work, 2023, 2025. So we've been identifying all these accidents, if you want, but it's telling us consistently that potentially not all the nutrients needed for an optimal embryonic development are there. And therefore, we need to be aware of that. And that includes minerals, vitamins, of course, amino acids, even linoleic acid, right? So does that mean that we should try to get them supplemented? Well, perhaps. Let's just start with the first window. I talked about two windows. The first one is broiler breeders. And we do know, we do know that broiler breeders, the nutritional status of broiler breeders will have an impact on the egg size, egg mass, and egg composition of fertile eggs. And therefore, that will have an impact on the development. And as a proof of concept, I have chosen one slide, the one that you have in front of you, which is from one of my PhD students, Bipul Chandra Ray, doing a great job. And without giving you a lot of details, because I don't want to spend time into the details, I want to spend time in the concepts today, right? So without spending time in the details, what we did is we tested 3 different levels of crude protein in broiler breeder diets, 16, 14, and 12. Now, all the diets were balanced. All the diets had all the essential nutrients, isoamine acidic, isoenergetic in metabolizable energy. So it should them all do good. And they actually did. Broiler breeders went really well with the 3 diets. They had no problems at all, even with the low crude protein diets at 12%. Their productivity was excellent. Their conversion ratio, excellent. Health, impeccable. Hatchability of the fetal eggs, precise, top notch. So what am I going to report? Well, I'm going to report something that it's the whole reason why I'm giving this presentation. The nutrient requirement of the poultry, of the broiler breeder, not necessarily is the same requirements that you will find in the embryo, because the developing embryos of the 12% crude protein diet, so the low crude protein diet, the developing embryos did not develop well. So the broiler breeders were perfect. The nutrition program was spot on, but not for the embryo. Are we forgetting the embryo? And the answer is yes, we are, right? So what happened with these embryos is that they couldn't develop well the gastrointestinal tract precisely. And I'm going to tell you why, because we did have a look at, we ran some transcriptomics, and we did have a look at what were what was the problem? So how come the gastrointestinal tract is not developing well? Well, first of all, the reason why the local protein diet impacted precisely the gut, it makes sense because the gastrointestinal tract, the digestive system, is rapidly developing. Is probably the most or one of the most, excuse me, demanding tissues in the developing embryo. Now the cell turnover is high, it needs to develop all the villi, etc., etc. So it is probably the most or one of the most demanding tissues. So if there's going to be a deficiency sign, that's precisely and probably going to be first being shown in the digestive tract. So it makes sense why we saw that. So what else did we see? The gene expression data really told us that it was all about the energy system. And I'm talking about in general. Of course, I could go gene by gene, and which I'm not going to do. But basically what happened is through peroxisomal and through mitochondrial deficiencies, the cells in the gastrointestinal tract, in this case was jejunum samples, didn't have sufficient energy to develop properly, and therefore the outcome was a smaller intestinal tract than the other treatments. Now, but I want to continue, and I want to talk a little bit in the last 15 minutes or so about this second window. I said, well, first window, proof of concept, we can change, we can improve, or we can negatively affect embryonic development through the broiler breeder, the transgenerational point of view. But I also want to talk a little bit about that second window. We have been working thoroughly. We've done quite a lot in Innovo. So we're taking that opportunity that many fertile eggs will be injected with the Marek's disease vaccine, and that happens during embryonic day 17.5 out of the 21. So this opportunity that we are already injecting vaccines, it's a fantastic opportunity that perhaps we could also inject at the same time some nutrients and some other functional supplements. And that's what I'm going to be talking about. How can we improve embryonic development through the Innovo techniques? Now, I do want to give credit to two friends who actually helped me a lot also in learning about Innovo techniques. And credit goes to Professor Firket from North Carolina State University, Peter Firket, and Professor Sehab Ayuni from the Hebrew University of Jerusalem. They are the two gurus who started all the Innovo feeding, and I appreciate the friendship and the learning that I had from them. But we've moved beyond that. Now here is a table, another busy one, and I promise you that's the last busy table that I have. But that comes from a review that I wrote last year, and it was about sort of summarizing all the nutrients, essential nutrients that when added in Innovo, they actually improve the development of the embryo. And you can improve it, as I mentioned before. We can target some of the systems that are particularly vulnerable or particularly relevant, important key systems. And in my view, if you ask me, those would be the digestive system and the immune system. And a number of different nutrients have a positive impact on developing either the immune system or the digestive tract. We have also looked at some metabolism. As you know, the developing embryo does consume a lot of oxygen, so there's a lot of oxidation going on in embryonic development. So having antioxidant impacts may also be beneficial, as it has been proven. Now, arginine, we have also tested arginine in our lab, and it does improve growth, lysine as well. Glutamine and glutamic acid, a bit less obvious, but branched chain amino acids. And in addition to proteins and amino acids, of course, energy could also be limiting. So you will see in the literature that there is positive impacts of energy sources, simple sugars, such as sucrose, dextrin, fructose, etc. Also fats, linoleic acid, but also vitamins and minerals. So again, and I think that's probably the main message of my talk, thinking that the fertile egg has all the nutrients that the embryo requires is probably not accurate to say. So we need to better understand the nutrient requirements of these embryonic beings to facilitate and to optimize their development. Let me explain you, though, a bit of an exercise that we did over the last 4 or 5 years. We've tested a number of functional ingredients, not only nutrients, as I was mentioned before, but also functional ingredients such as essential oils. And we've published that, and these are available. Now, targeting in principle 2 main systems, as I mentioned before, the immune and the digestive system. How do we do that? We inject excuse me, in the amnion at day 17.5, and we know why it's at day 17.5. And the amounts, well, we tested different amounts above 10 microliters of carbacril, which is a model essential oil that we tested. Above 10 microliters is toxic, so there is a negative impact on hatchability. But up to 10 microliters, we start seeing positive impacts of different essential oils. We selected 27 essential oils, of which 5 happened to be toxic to the embryos, and we discarded them, of course. So we progressed our research with 22 essential oils. So I'm going to be showing you some of the impacts of these 22 essential oils on gene expression, that these genes being related to either the digestive system or the immune system. The first group of genes that we looked at and that we had positive impacts were associated to gut integrity, and you would all be familiar with occludin and zonula occludins 1 and 2, claudin 1, Marvel domain contain, and then the epidermal growth factor receptor, or the epidermal growth factor itself, EGFR and EGF. Now, if you will look at the 22 essential oils that remain in our pipeline, you will see that claudin 1 is particularly positively impacted by a number of these essential oils: black pepper, clary sage, a bit less lemon myrtle, but a bit, but also turmeric acid, ylang ylang. And you will see that different genes are affected by different essential oils, as you may imagine. So different essential oils have different modes of action. Yeah, clear cut. Now, on the right side of your slide, you see the correlationship. And all these genes that were very specifically selected are related because they are part of the gut integrity biomarkers. And as you see with the asterisk, they are highly correlated, positively correlated, which means excuse me, that if we improve one, it is likely that we are also improving the other gut integrity biomarkers. This was, by the way, a work done by my then student, now Dr. Asad Ali Kashkeli, who is from Pakistan. He's currently in the United States. Now, having a look at some other digestive system biomarkers, we chose amino acid transporters, EA83, PEPT1, also acid binding protein, fatty acid binding protein, sodium glucose transporter, and then the aminopeptidase and the sucrose isomaltase. Now, again, I'm not going to go through all the 22 essential oils, but you would see that some are highly improving the expression of these biomarkers. Again, most of them would be highly correlated. They are, if one is doing better in some enzymes and transporters, is also doing better with the others. So they go in a pact, and that's why we put them together, in fact. Now, the other aspect that I was particularly interested was the immune system, as I mentioned. So let me show you some of the results on the immune system. First, as a background, you would probably know, it's needless to say, that really the immune system, it develops fairly quick in the embryonic animal, in the embryo. And as you see, very early, you start having some cytokines circulating in the embryo from day 0 to 5 of embryonic stages. But certainly by the time we usually have our Innovo interventions, day 17.5, the immune system is fully active, fully developed. That is either with functional TMB cells, but also you'll see the cytokines. So there you go. So we go and start injecting our essential oils, and you see a number of impacts on selected genes that we look at their expression, including some cytokines, 1 beta and interleukin 10, and some immunoglobulins M, the CD factor, etc. So as you see again, and I'm not going to go through each one of the essential oils that we tested, but you see that some of the essential oils are particularly interestingly improving that expression or activation of the immune system, black pepper, star anise, ylang-ylang, etc., etc. Some of these we have already mentioned a couple of times. Now, in addition to that, we were interested in looking at, all right, so we're injected during the embryonic stage, but is there any impact post-hatch when we inject it during the embryonic stage? And we looked at it, we've done a few experiments. I'm going to show you just one very quick, because we have quite consistent results when it comes to the immune system. So we inject the embryo with carvacrol, which is the main active compound of oreganum essential oil, embryonic day 17.5, and we looked at the gene expression and the impact on cytokines up to day 14. Now, when we looked at the transcriptomic data, the overall main pathways that we see activated are related to passive immunity, that is, antimicrobial peptides being released, or defense responses against gram-negative bacteria through CCR6 chemokine receptors, etc. So it is true that we can start activating the immune system at an early embryonic stage, and that, in our opinion, may be fundamental to get that chicken post-hatch ready to go. So we're starting to activate the immune system early enough so that the post-hatch, we give them some additional robustness. And that is somehow confirmed by the fact that we still see the effects at day 14 post-hatch, right? Still we see that IL-1 overexpressed, IL-10 is overexpressed at day 14, even though carvacrol was administered during embryonic stages. So, and I'm finishing up with a bit of a summary of, I intended to give you a journey through that transgenerational nutrition that, in my view, is actually giving us an extraordinary window of improving the life of the chicken, the robustness of the old chicken, but also the understanding of how these systems develop. And as I mentioned in my first slide, there's two main windows that we currently are investigating to improve that robustness of day one chick. And the two windows are, one, truly transgenerational through broiler breeders, and try to make sense of the nutrients that we need to deliver through the breeders to optimize egg mass, egg composition from an embryonic point of view, not from a breeder point of view, but from an embryonic point of view. We do have also a shortcut that we call Innovo feeding or Innovo supplements, where allows us to supplement embryonic development with essential nutrients that effectively will improve physiology, metabolic changes through epigenetic mechanisms as well, of course. But we have particular interest, as you would guess, to improve the immune response and the digestive activity. And with that, I will finish by acknowledging the three students who were able to give me that data to present. Bipul Chandra Ray, who's currently a PhD student finishing, and Dr. Mila Meyer and Dr. Asad Kashkeli, who already finished last year. Congratulations to them. They all are extraordinary students, and I want to thank them and give them my acknowledgement. And also the funding bodies. Basically, these projects were funded by the AgriFutures Australia Chicken Meat Program and the Consortium Nutrition, Gut Health and Environment. And with that, I'm going to stop sharing and happy to see if there's any question. I think I'm almost running out of time. Sorry for extending my presentation. Thank you so much for your attention.
Thank you so much, Dr. Rora, for such a fascinating insight into embryonic development and its potential role in improving the poultry production. While the audience is going to write a question, I'll kick off with one question. It's a very fascinating area of work that you have presented. And despite the fact that in novo supplementation has shown potential, great potential, and we know that it works, my question is that despite these advantages, commercial adoption remains relatively limited. What do you think is causing this limitation, and why the industry is not adopting it? Because we know the benefits.
That's an excellent question. Thank you so much, and thanks for your good comments, Professor Katak. I appreciate that. Well, in my view, there is a risk in injecting additional nutrients to the developing embryo. There had been very little work done on compatibility of nutrients and other active functional compounds. How are they compatible with the actual Marek's disease vaccine, for example? Because that's fundamental. We have developed the concept of in novo feeding and the concept of in novo supplements by saying, all right, we're going to take the same window than the Marek's disease vaccine so that there's only one injection when it comes to the point. But then I'm not sure about you, but I haven't seen many reports of actually looking at how different compounds may impact the efficacy of the Marek's disease vaccine. And that's been, in my opinion, a miss. So as you would know, commercial producers, they are very aversive to changes and they will not risk. So we need to make sure that we have sufficient data that consolidates the knowledge on in novo feeding application, and particularly in the case of administering those compounds together with the vaccine. That is a crucial aspect. The other aspect, in my view, if I may add, is probably consistency. You look at the literature and there's not always consistency in the results of some of these injections. But the reason of the lack of consistency is because of the procedure. I was lucky enough to be trained directly by my colleagues Sehava and Peter, Professor Sehava Uni and Professor Peter Furquet. And I think we are excelling in doing the technique, but it's not a given. You have to really target the amnion, and that's easier said than done. So having the expertise of labs such as Furquet and Uni's lab is also fundamental to have consistent results. And I think that's probably the package that needs to improve overall, and that we're trying to do.
Thank you. Thank you very much. That was, I do agree, because I think there is a lot of potential, but as you said, easy said than done. It's the technique which makes it a bit difficult for people to adopt it very easily, and especially at the hatchery site. If this can be adopted in future, I think that will be a big revolution there. So once again, thank you very much, Professor Arora, for joining us and for your very valuable talk. Thank you so much.
Thank you so much, and congratulations for such a nice event.
Thank you.
Thank you very much, Professor Eugenie Rora. As a small token of our appreciation, we would be delighted to recognize your contribution during the session closing, and a certificate of presentation will be awarded at the end of the session. So now we request you or ask you, if your schedule permits, we would be honored if you could remain with us until the end of the session. However, we fully understand your other commitments as well. And if you need to leave earlier, we will be pleased to share your certificates via the email.
Thank you so much. I'll try to stay, and thanks for the appreciation. It's nice of you.
Thank you.
Thank you.
Thank you very much. So do we have the next speaker online?
Yes, Professor Oğuz Yılmaz from Ankara University, Turkey. She is on board.
Oh, right. Okay. Very welcome, Professor Oğuz Yılmaz. She is from University of Ankara, Turkey. Professor Suzy Maz focuses on poultry nutrition, feed evaluation, alternative feed ingredients, and sustainable feeding system. Her research contributes significantly to improving nutritional efficacy while supporting environmentally responsible poultry production. Her presentation today is entitled From Feed Formulation to Gut Programming. Please join me to welcome Professor Suzy Maz. Welcome, Professor Suzy Maz, and mic is yours. Thank you.
Thank you, too. Let me share my slides. Yes.
Yeah, we can see your slides. Thank you.
Okay, good. So, no. Okay.
Can you? Yeah, it's now
It's
on
okay?
presentation mode. Yes, thank you. Yeah.
Good morning, everyone. It's a pleasure to be here with you today. Thank you for the invitation, of course. Actually, before I begin this presentation, I would like to ask a simple question for the feed formulation. How many of you think nutrition is mainly about formulating feeds and meeting nutrients requirements? This is the basic questions. Because the feed formulation is not all paradigm in the feed formulation and the ration and the nutrition for the broilers or poultry. For decades, that has been exactly how we approached animal nutrition. We focused on energy, protein, amino acids, vitamins, and minerals. We calculated requirements, balanced rations, and optimized feed formulation. That's okay. And to be fair, this approach has been tremendously successful. Modern poultry production has achieved remarkable improvements in growth rate, feed efficiency, and productivity. Birds today reach market weight faster and more efficiently than ever before. However, despite all these advances and puzzling questions remain. Why do, for example, flocks receiving exactly the same feed raised under very similar management conditions often show different performance outcomes? Because this is biological intelligence, we can say. Why do some flocks achieve exceptional FCR while others fall behind? Why do health challenges still emerge despite increasingly sophisticated nutritional programs? The answer may not lie in the feed itself. The answer may lie inside the bird. Today, I would like to introduce you to a concept that is transforming modern animal nutrition. This is biological intelligence that I mentioned. This concept challenges the traditional view of nutrition and suggests that feed is not simply fuel. Feed is information. Feed acts as biological software capable of programming how the gut develops, how the immune system responds, and ultimately how efficiently an animal performs throughout its life. As future veterinarian, as you, understanding this shift will be critical because the next generation of animal production will not be driven only by nutrition, genetics, or management. It will be driven by our ability to understand and program biological systems and bioavailability digestion. To understand where we are heading, we first need to understand where we came from, of course. Traditional nutrition was largely based on deficiency prevention. The primary objective, we simply provide sufficient nutrients to support growth and prevent disease, of course. Nutritionists relied heavily on nutrient requirements tables focused on ensuring that energy, amino acids, vitamins, minerals met the animal requirements needs. This approach worked extremely well when productivity was limited by nutrient deficiency. However, today's animals are different. Modern genetic selection has dramatically increased growth potential and production efficiency. As a result, nutrition is no longer simply about preventing deficiencies. Instead, nutrition has become a tool for directing biological responses. Every ingredient entering the gastrointestinal tract delivers molecular signs. For example, these signs influence the gene expression, cellular metabolism, immune response, immune development, barrier integrity, and microbial composition that is called the microbiota. In other words, nutrition is no longer only about supplying nutrients. Nutrition is about influencing biological decisions. This shift is what we call precision nutrition. And this paradigm shift represents one of the most exciting developments currently occurring in animal science. Actually, here. So, what exactly do we mean by biological intelligence? Traditionally, the gastrointestinal tract was viewed as a relatively passive organ. Food entered, nutrients were digested, absorption occurred, waste was eliminated. Today, we know this view is far too simply, very simple. The gut is actually one of the most sophisticated sensing systems in the body. It continuously monitors nutrients, microbial metabolites, toxins, pathogens, and environmental signals. Based on these inputs, it adjusts its structure and function in real time. Think of the gut as a biological computer, maybe we can. And every meal provides data for us. Every microbial metabolite provides feedback, and every immune signals influence feature responses for us. The gut processes this information and determines how resources should be allocated. Should energy be directed toward growth, toward immune function, immune defense, or toward tissue repair, or maintaining homeostasis? This ability to sense, interpret, and respond to biological information is what we describe as biological intelligence for the body. And perhaps most important thing, this intelligence can be influenced through nutrition, from feed to the body or gut health maybe. That means veterinarians and nutritionists have the opportunity is not only to feed animals, but also to shape how biological system function. One of the biggest challenges in understanding biological intelligence is measuring it. Historically, we evaluated nutrition using performance indicators such as, body weight, daily gain, FCR, feed conversion ratio, and the mortality, maybe the carcass parameters. These measurements are useful, of course, but they tell us what happened after the fact. They don't explain why it happened. This is where multi-omics technologies become extremely valuable. Genomics helps us understand genetic potential, or transcriptomics tell us which genes are being activated or suppressed. Metabolomics reveals what is happening metabolically in real time. Together, these technologies allow us to observe biological response at the molecular level. Instead of waiting until performance declines, we can identify metabolic disturbances long before clinical signs become visible. This represents a major shift from reactive management to proactive management. These technologies help and will likely become increasingly important tools for decision making and, of course, the health monitoring. Among all omics technologies that I mentioned, metabolomics may be one of the most practical. Why? Because metabolites provide a direct snapshot of what is happening inside cells at a given moment. Imagine looking at the dashboard of a car. You can immediately determine whether fuel levels are adequate, whether the engine is overheating, or whether a warning light has appeared, Metabolomics provide a similar dashboard for this biological system. For example, reduced plasma glutamine levels often indicate that the gut is under stress condition. Elevated ammonia and uric acid levels may suggest inefficient protein utilization and unnecessary energy. These changes can occur days or even weeks before performance losses become obvious. And this gives producers and veterinarians a valuable opportunity to intervene early. Instead of responding to problems, we can predict and prevent them. And that is one of the central principles of precision nutrition. Now that we understand feed as biological information, an important question emerges. When should programming begin? Most people would probably answer after hatching. However, modern research suggests that it may already be too late. The foundations of future performance are established during embryonic development. The embryo is not a passive structure waiting to hatch. It's a rapidly developing biological system that is already responding to nutritional and environmental signals. This concept is known as early gut programming or in-ovo feeding, Through in ovo interventions, we can influence intestinal development before the chick even enters the outside world. One example is sodium bitrate for administration during incubation or any feed additives maybe. Studies have shown that such interventions accelerate intestinal maturation, improve villus development, gut development, and enhance immune responses. What makes this particularly fascinating is the concept of development plasticity. Small interventions during critical development windows may create long-lasting effects that persist throughout the bird's life. In simple terms, we are preparing the biological hardware before the operating system is switched on. For the future veterinarians, this represents an entirely new way of thinking about preventive medicine and animal production, of course. The moment of hatching represents one of the most critical biological transitions in an animal's life. At hatch, the gastrointestinal tract is rapidly colonized by microorganisms. These pioneer microbial communities play a major role in determining feature for the animal's gut health. And we often focus on the feed composition. However, timing may be just as important. Delayed access to feed and water can significantly influence microbial succession, Research shows that prolonged delays may favor opportunistic bacteria such as E. coli maybe or Shigella species. In contrast, immediate access to nutrients encourages the establishment of beneficial microbial populations and other health-promoting organisms. Why is this important? Because microbes don't simply live in the gut. They actively participate in gut development. They influence epithelial maturation. They regulate immune function, immune education. They produce signaling molecules, and they help shape the animal's physiological trajectory. And in many ways, the microbiota can be viewed as the animal's feed lifelong biological partner. Let us now move to one of the most important concepts in modern gut health, barrier integrity. The intestinal barrier serves as a highly selective interface between the external environment and the internal body. Its job is surprisingly complex. It must allow nutrients to enter efficiently. At the same time, it must prevent pathogens, toxins, and inflammatory molecules from crossing into circulation. When this barrier functions properly, performance remains high. This is increasing. When it fails, serious consequences follow. Many of you have heard the term, for example, the leaky gut. This condition occurs when intestinal permeability increases beyond normal physiological limits. As a result, harmful compounds gain access to systemic circulation. The immune system becomes activated, inflammation increases, nutrient utilization, of course, decreases, and feed efficiency declines. From a production perspective, barrier failure represents both a health problem and economical problem. Every unit of energy directed towards managing inflammation is a unit of energy that cannot be used for growth. That is why maintaining barrier integrity has become one of the central objectives of precision nutrition. If the intestinal barrier is a fortress, then tight junction proteins are the gates. Among these proteins, two are particularly important, zonal occludins and occludin. This ZO1 acts as a structural scaffold that maintains cellular organization. Occludin functions as part of the sealing mechanisms between neighboring epithelial cells. Together, they regulate paracellular permeability. In other words, they determine what can and cannot pass between intestinal cells. When expression of these proteins decreases, permeability increases. Then expression increases, barrier strength improves. What makes this exciting is that nutrition can directly influence these genes. Certain dietary interventions have been shown to upregulate these genes' expression, thereby strengthening intestinal integrity. This means that feed ingredients are not simply providing just nutrients. They are actively influencing the architecture of intestinal barrier itself. And building a strong barrier is only half the story. We must also maximize nutrient uptake. Imagine two birds consuming identical diets. One absorbs nutrients efficiently and the other absorbs them poorly. So performance outcomes will differ, of course, dramatically. This is where transporter proteins become important. For example, peptide 1 is responsible for transporting the peptides and the tripeptides. OCGLT1 is responsible for glucose transport. These proteins function as molecular delivery system. The more efficiently they work, the more efficiently nutrients enter the circulation. Bioavailability. Precision nutrition therefore focuses not only nutrient supply, but also on nutrient utilization, bioavailability in the body. The goal is not simply to place nutrients in the intestinal lumen. The goal is to ensure that nutrients successfully reach the cells that need them. This represents a fundamental change in nutritional thinking. We are moving from feeding nutrients to optimizing absorption system. So far, we have discussed intestinal barrier and the proteins that maintains its integrity. However, before pathogens even reach this barrier, they encounter another protective layer, mucus. This mucus layer is not simply a slippery coating. It's an active component of the immune system. The major structural component of this layer is MUC2, a mucin protein secreted primarily by goblet cells. Think of mucus as a biological buffer zone. It creates physical distance between microorganisms and epithelial cells in the gut. The greater this distance, the lower the likelihood of direct pathogen attachment and the invasion. This is important because many intestinal pathogens must first attach to epithelial cells before they can cause disease. If they cannot reach the target, they cannot establish infection. Dietary fibers, phytogenic compounds, and certain microbial metabolites have all been shown to stimulate this gene expression. As a result, nutrition can directly influence one of the body's most important defensive system or the structures. This is another excellent example how feed acts not only as nutrition, but also a regulator of biological function. Yes, some feed additives affect this. For example, phytogenic modulators. Let us examine practical. The first strategy involves phytogenic compounds. Phytogenics include plant-derived bioactive substances such as essential oils and botanical extracts. Examples include cinnamon oil, cinnamaldehyde, team extra, something like that. Initially, these products gained attention because of their antimicrobial effects. However, their biological influence extends much further. Many phytogenic compounds affect digestive enzyme activity. Others influence immune responses. Some even regulate gene expression associated with nutrient transport and the barrier integrity. This makes phytogenic particularly attractive in antibiotic-free production system. Instead of simply killing bacteria, they help create a more balanced and resilient intestinal environment. Their role illustrates a key principle of biological intelligence. The most effective interventions often work by supporting biological systems rather than fighting against them. And the other feeding strategy as a feed additive is the microalgae. These are among the most exciting emerging ingredients in animal nutrition. One species receiving considerable attention is, the chlorella. Microalgae contain a remarkable range of bioactive compounds, including polysaccharides, pigments, antioxidants, vitamins, and bioactive lipids. Research demonstrated improvements in villus height and the ratio of the villus to crypt following the supplementation. Why does this matter? Because greater absorptive surface area increases nutrient utilization effectively. In addition, microalgae have been associated with improved expression of barrier-related genes such as ZO or occlusion, as I mentioned. This means that they simultaneously support absorption and the production. From a biological intelligence perspective, microalgae function as multifunctional regulators rather than simple nutrient source. They influence multiple physiological pathways at the same time. This is very well known, you know, another strategy, prebiotics, probiotics, and the postbiotics, maybe symbiotics. Perhaps no area nutrition has expanded more rapidly than the microbiome modulation. Prebiotics provide selective substrates for the probiotics or the beneficial microorganisms. Probiotics introduce beneficial living microorganism. Postbiotics deliver beneficial microbial metabolites and the cellular component. Together, these approaches create a more favorable intestinal ecosystem. One particularly exciting aspect of postbiotics is that they don't require living organisms. Instead, they deliver the biological signals generated by those organisms. This can improve stability and consistency under commercial conditions. As our understanding of microbial host interactions grows, microbiome-directed nutrition is likely to become increasingly precise. In the future, diets may be designed not only for the animal, but also for the microbial communities living within it, maybe just like an ruminants.
I'm sorry, Professor Sismaz, may I
Yes.
kindly inform you that you have only 2 minutes left for your presentation. Can you please wind up?
Thank you. So I need to pass maybe here. What do you think? Just 2 minutes, you said?
Yeah.
Okay. At the end, maybe I can continue from here. For the future vision, you know, everybody knows the AI and the precision nutrition. There are some relationship between them. So can we look this towards as the future? Many of you sitting here today, you know, will practice veterinary medicine in the world veterinary different from the one of your professional entered. Artificial intelligence is already transforming healthcare, agriculture, and biological research. Animal production will be no exception. The amount of biological data generated today is enormous. Genomic data, microbiome data, metabolomic data, environmental data, performance data. No human being can process all of this information simultaneously. But nowadays, artificial intelligence can. It's a little bit complicated. Deep learning systems are already capable of identifying patterns that remains invisible to human herbs. Many of students attending this congress I know today may become the professionals who bring these technologies into everyday practice. So this is a little bit complicated, but it's very important point. And as we come to the end of the presentation, I would like to leave you with one final thought. When I began studying animal nutrition, feed was often viewed as a collection of ingredients, just ingredients, corn, soybean meal, vitamins, minerals, something like that. Today, we understand that feed is much than these feed materials actually. Feed is information. Feed is communication, feed is biological programming. We are no longer simply feeding animals. We are influencing how genes are expressed, how immune systems develop, how microbiota assemble, how barriers function, and ultimately how performance is achieved. This means that the future of your, maybe the profession extends far beyond traditional boundaries. The next generation of veterinarians will not only diagnose disease, they will interpret biological data, they will work with artificial intelligence, they will understand microbiomes, they will integrate nutrition, immunology, molecular biology, and the data sciences in many ways. So if there is one message I hope you remember from today is this: we are no longer simply feeding birds. We are writing the biological code maybe to determine their resilience, efficiency, health, and the future. And perhaps even more importantly, you will be among the professionals writing that code. Thank you very much for your attention. I would be happy to take your questions. Thank you.
Thank you very much, Professor Sumas, for your very detailed presentation on a very current topic of metabolomics and trichomonomics, or all these kind of markers which are really getting a lot of attention. We are really running out of time, but I have a question and I have put it in the chat. So if you have time, do answer, because I do sometimes wonder. We do all these kind of interesting marker analysis and everything, but in reality, it never come into back to the poultry industry, and we never have anything which can translate into the industry from our studies. What is the bottleneck there? But I'm afraid we are running out of time, so I will leave that discussion. I'll leave that question on the chat, and if you have time, please share your thoughts on that. And I would really like to thank you for your a very thought-provoking presentation. Thank you so much.
Thank you too.
Yeah, with this, we will now move on from our key presentations to two of our emerging scientists whose work reflect the innovation and international collaboration that this conference seeks to promote. First among them is Dr. Abdul Ghayas. Dr. Abdul Ghayas is from Islamia University of Bhawalpur, Pakistan, and he is going to present his talk on interactive effect of genotype and production system on nutritional quality and fatty acid profile of fast and slow-growing chicken. I hope Dr. Abdul Ghayas is there. Please join me to welcome Dr. Ghayas.
Yes, Dr. Abdul Ghayas is on the board. Can you hear us, Dr. Abdul Ghayas?
Can you listen me, sir?
Yeah, we can listen to you. Please, can you share your presentation?
Yeah. Just 1 minute.
Dr. Ghayas, before you start your presentation, just to remind you that you have only 10 to 15 minutes for your presentations.
Okay, sir. Okay, sir. Thank you so much.
Yeah, we can see your presentation. Please change it to the presentation mode and you're good to go.
Presentation mode, yes. Just 1 minute.
Yeah, perfect.
Is it okay now?
Yep, now perfect. Thank you. Please, all yours.
Yes, thank you, madam. My name is Abdul Ghayas, and I'm working as assistant professor in the Islamia University of Bahawalpur. My topic of presentation is interactive effects of genotypes and production system on nutritional quality and fatty acid profile. This is basically my PhD thesis research. And
excuse me, Dr. Abdul Ghayas.
Yes, sir.
Can you please open your camera?
Yes.
Are you able to open your camera? Okay, now. Thank you. Thank you so much.
Okay, sir. This research was basically part of my PhD research, and this research was conducted on University of Veterinary and Animal Sciences in the Department of Poultry Science. This research, sorry, just one time, I have a problem. I think slide is not going, not moving.
You can share your slides again.
Yes.
No, it's working now.
Yes. Poultry meat is one of the most consumed item in the world, and we have basically the a variety of consumer. Some consumer likes thigh meat, some consumer like breast meat, and in today's market, consumer are increasingly prefer the meat from meat with a better nutritional profile, with low fat profile and healthier lipid profile. And for these, production system and genotype can play a significant role. Especially the free range system may influence the activity level and access to the forage and oxidative status, and ultimately the fatty acid profile of the meat. Similarly, genotype may have a significant impact. In this research, our research question was which genotype and production system combination may produce best meat with the better proximate composition, stronger antioxidant profile, and healthier fatty acids in the meat. And our aim of the study was to evaluate the effect of genotype and production system on these parameters. We hypothesized that slow-growing genotypes, particularly the Rhode Island red, reared under free-range system, will produce nutritionally superior meat in terms of proximate, in terms of lipid content, and fatty acid composition. For this, we applied 3/2 factorial arrangement of 720 day-old chicks. These 720 day-old chicks were from 3 genotypes, Cobb 500, Cobb Sasso, and Rhode Island red. And these birds were distributed in two production system, intensive and free range. And free range basically was provided after 21 days of age. Before 21 days, all the birds were intensively reared. After 21 days, they were given, they were distributed into two production system, while the sampling of the meat were carried out at 56 days of the age. And we evaluated the main effect of genotypes as well as the production system and the interaction of both genotype as well as production systems. In proximate, we evaluated the dry matter, crude protein, ether extract, and ash content. And in antioxidant status, we evaluated the total phenolic contents, total flavonoid, and thiobarbituric reactive substances as lipid oxidation markers. And fatty acid analysis were also carried out. We also evaluated the saturated fatty acid, unsaturated fatty acid, polyunsaturated fatty acid, and among those, N6 and N3 fatty acid. And when we got the result, production system and genotype both have significant impact on the proximate composition. Especially the free range bird have higher crude protein, higher dry matter content, and lower ether extract. Almost 0.85 points of dry matter and 0.85 points of crude protein were higher in the free range birds. Similarly, ether extract and ash content were also significantly influenced by free production system and genotypes. In free range, the ether extract was lower as compared to the intensively reared bird. Ash content were higher in the free range bird also. Among the genotype, Rhode Island birds were much superior, such as it contained the crude protein content of 24.7%, and ether extract was also better in slow-growing bird, especially the RIR and Cobb Sasso. And these both slow-growing bird, Cobb Sasso and RIR, were non-significantly different in ether extract. On overall basis, slow-growing genotypes presented the better proximate profile. This was an expanded view of these results. In terms of oxidative stability, free range increases the antioxidant content in the meat. And just 1 minute, sir. Free range increase the total phenolic as well as total flavonoid content, while the thiobarbituric reactive substances were decreased in the free range systems, indicating lower lipid oxidation. However, when Rhode Island red bird were reared in the free range, they produce strongest antioxidant profile, presenting 28.8 milligram units of total phenolic content and 3.84 of total flavonoid content and 0.09 point of thiobarbituric reactive substances, which were more favorable as compared to the other interactions. These were the major results when results were compared with the different treatments. In terms of fatty acid profile, production system and genotype both had significant impact on the fatty acid profile, especially the healthier fatty acids such as oleic acid and linoleic acid and arachidonic acid. They were increased under free range condition, indicating a shift towards a healthier unsaturated fatty acid profile. Both arachidonic and alpha-linoleic acid were higher in the free range. Alpha-linoleic acid, when increased, it supported the improvement in the total N3 fatty acid. And these parameters look compressed in the mixed scale chart when I presented on the left side. Slow growing in terms of genotype, Rhode Island red chicken were further better. It presented the higher monounsaturated fatty acid and polyunsaturated fatty acid as well as total N3 values. When the N6 N3 ratio is calculated, it basically shows the healthier value, which also comes under Rhode Island red in the favor of Rhode Island red chicken. Rhode Island red and Cobb's also had higher total N3 fatty acid as compared to the Cobb 500, which Cobb 500 basically was the commercial fast growing bird. Cobb's also basically were the commercial slow growing, and RIR we considered it as rural slow growing. In all these parameters, commercial rural slow growing RIR chicken showed the lowest N6 to N3 ratio, which is nutritionally the most favorable pattern for a healthy meat. Free range in terms of interaction, free range and in Rhode Island red chicken one reared under free range condition. This combination produced the most favorable meat quality profile and presented these values: 0.99% of ether extract and TPC value was 28.84, T-bar value was 0.09, and ultimately the N6 to N3 ratio was 11.53. And this is the basically a heat map chart for the fatty acid profile. Darker green indicated a more favorable value after the column-wise normalization. N6 N3 ratio lower values are more favorable, and ultimately the Rhode Island red and free range combination, it showed the strongest overall profile of this research. Now, why the free range or RIR reared under free range produced the most favorable meat quality? It was basically due to the slow growth biology, lower growth pressure, and different lipid deposition pattern. And metabolic effects also played a significant role, such as lower fat accumulation and improved oxidative stability has been observed in the slow growing bird, and that ultimately produced the meat with superior quality. And this research has also the practical applications. Slow growing genotype, we can recommend the farmer to raise a slow growing bird optimum for healthier meat production. And this strategy is that we identified that RIR in the free range can be a natural strategy for the production of healthy meat without relying on the post-harvest interventions. In this study, our main conclusions were both genotype and production system significantly influence the meat quality traits. Free range rearing increase the dry matter, crude protein, antioxidant compound, and beneficial unsaturated fatty acids, while reducing the ether extract and thiobarbituric reactive substances. The Rhode Island red chicken showed superior nutritional traits compared to the COP 500, COP Soso, and the combination of Rhode Island red and free range produced the best overall meat quality profile. From this, you can ask any question. Does anyone have any questions, sir?
In the chat box, there is no question right now. And thank you very much, Abdul Ghayas, for such a wonderful presentation. Dr. Farina, it's over to you, ma'am.
Yeah, thank you very much. Thank you, Abdul Ghayas, for a very nice presentation. However, I have a quick question before we go on to the next speaker. My question is that you compared two very different purposes breeds, although both are slow growing, but both have different purposes. One is dual and one is, how did you manage the production period? What was the production period for your comparison?
Yes, your question is valid, ma'am. Basically, this study was not a single experiment study. We also have compared the weight-based traits. In this study, we only have production system and genotypes. Then we have compared the weight-based.
Yeah, no, but what I mean is for the study, the data that you have presented,
yes,
where you show that there was a reduction in T-bars in case of RIR. What was the duration of the trial? Was it the full, like 35 days, 59 days, 65 days? What was the duration of the two trials that you compared?
Yes, the overall
Let's
study.
take one parameter. Let's take one parameter of T-bars.
Yes, overall, this study was comprised of more than 1 year time period.
Okay.
Yes.
Okay, thank you. Thank you very much. Yeah. Thank you. I don't see any question in the chat box. So, and because we are also aware of the time. So thank you very much, Dr. Ghayas, for sharing your PhD data and your result. I can see that it's a huge amount of data and difficult to squeeze everything in within the short time that was given to you. But it was a fantastic presentation. Thank you so much for that. Our next presenter is Muhammad Omar Asghar. And this is our final presentation for this session. And Muhammad Omar Asghar is from Warsaw University of Environmental and Life Sciences, Poland. His presentation is on hydrobarothermal processing as an engineering approach for enhancing the functional properties of non-GMO soybean meal in poultry nutrition. I would like to welcome Muhammad Omar Asghar. I hope he is online and he can share his camera. Yeah, thank you very much, Muhammad Omar. The mic is yours, and if you don't mind sharing your presentation, and then you can start from there. Thank you.
Just to mention before Omar's presentation, Omar is serving the PPS board. He's our Secretary General of PPS from Wrocław University of Environmental and Life Sciences, Poland. So it's over to you, Omar. You have 10 minutes for your presentation. Please wind up as soon as you can.
Okay. So good morning, everyone. Thank you so much for giving me this opportunity to discuss my preliminary results from my PhD result here in this prestigious poultry nutrition conference. As you know, my name is Muhammad Omar Asghar, and I'm a PhD research scholar at Wrocław University of Environmental and Life Sciences, Poland. Here is the topic of my presentation. So here I have shared the presentation plan. I will discuss briefly about the background and problem, hydrobarothermal processing technology, objectives and hypotheses, material and methods, and key results, and take-home message and conclusion at the end. As you know that the world population is increasing day by day. To cope up the protein requirements of the world, poultry meat is the best and cheap alternative protein source as compared to other red meat sources. As you know that the poultry sector will make a substantial contribution to the food security and nutrition for providing minerals, protein, and essential oils in our diet. Before we dive deeper into the detail, I will briefly discuss the importance of soybean in poultry. Soybean are a global nutrition powerhouse serving as a vital source of protein and oil in our diets. It contains balanced concentration of amino acid in it, and the crude protein content contains 44 to 48%. The problem is that we are facing several challenges nowadays in poultry sector. For example, the Polish government is going to ban on the use of GMO soybean in all animal diets. And the second problem is about, that the European Union is not self-sufficient when it comes to protein production. So most of the time we have to import this soybean from other countries like Brazil, Argentina, and USA. So in this way, we have to spend a lot of money on the transportation to import this feed ingredient here. And the third problem is related to the processing of soybean. For example, both over-processing and under-processing of soybean leads to alter the nutrition profile of soybean. I will discuss in detail in my upcoming slide that how over-processing and under-processing of soybean leads to alter the nutritional profile of soybean. As we know that the quality of soybean basically depends on two factors. The first one is the concentration of amino acid present in it. And the second one is the level of chemical components known as anti-nutritional factors it contains. As raw or unprocessed soybean contain different levels of anti-nutritional factors. For example, protease inhibitor causes pancreatic hypertrophy, which ultimately leads to poor growth performance in the birds. And the second one is the trypsin inhibitor also causes reduce in the protein digestibility and amino acid bioavailability to the birds, which ultimately disturb the gastrointestinal microbiota in the birds. Here in this slide, you can see that we must completely inactivate the anti-nutrition factor without degrading the protein quality. For example, we have to reduce this anti-nutrition factor. So for this, we are using conventional thermal processing technologies. I will discuss in my upcoming slides that what kind of methods we are using. And the second problem is the over processing or overcooking or heat extra providing, which can ultimately reduce the protein quality or destroy the amino acids structural integrity needed for the growth in birds. Here in this slide, you can see these are the conventional methods to reduce anti-nutritional factors, includes soaking, germination, roasting, and fermentation to reduce these anti-nutritional factors in soybean meal. Each method have its own pros and cons. And in this slide, you can see that there are four major conventional processing technologies, which includes heat treatment, ferment treatment, and chemical treatment and enzymatic treatment to reduce these anti-nutritional factors in soybean. And these methods which ultimately reduce the anti-nutritional factors, but at the same time, it also degrade or reduce the protein structure of soybean. And this is the major problem while processing of soybean called Maillard reaction. This is basically a reaction between amino group of amino acids and the reducing sugar to form melanodines. And this reaction usually starts at 140 degrees Celsius temperature. And some of the scientists are saying that these Miller reactions are beneficial. We can easily control by temperature, time, and water moisture level. And you know that Miller reaction produce some melanodynes, which have antioxidant properties and anti-tumor effects. And on the other side, if we are talking about the negative effects of this Miller reaction, this causes loss in the heat-sensitive vitamins like vitamin A, B, and C, and it also produces some carcinogenic compounds at high temperature like acrylamide. And here in this slide, I have briefly described this innovative hydrobarothermal processing technology to reduce anti-nutritional factors and as well as improve the protein quality of soybean. In the first step, the crushing of the soybean, and in the third step, conditioning, we are providing 95 degrees Celsius temperature. And in the fourth step, here is the, you can see here is the innovation because we are providing temperature, time, pressure. At the same time, we are also providing moisture just to protect the protein quality of soybean. Otherwise, you know that at high temperature, Maillard reaction will start and ultimately leads to denaturation of protein quality. And we want to protect this protein quality in soybean. That's why we are working on this hydrobarothermal processing technology. So here we have given them different time, temperature, and pressure to produce 96 different soya prototypes. I will discuss in the upcoming slides about the result of this processing technology. And here in this slide, I have briefly differentiated between these conventional thermal processing technology and this hydrobarothermal processing technology. As you can see here, the first point is the heat reliant. In the conventional thermal processing technology, we must take care of it because in the hydrobarothermal processing technology, as I mentioned earlier, that in this we can stop this Maillard reaction. And the second point is the anti-nutritional factor. In the conventional thermal processing technology, we are unable to achieve or reduce this anti-nutritional factor less than 3 milligram per gram of the feed ingredient. But in hydrobarothermal processing technology, we are able to achieve this target. And the third one is the protein quality and amino acid digestibility. We are able to achieve this in this processing technology as compared to conventional processing technologies. And here's the hypothesis that hydrobarothermal processing technology produce experimental soybean product. And our hypothesis regarding this product is that this can increase the protein digestibility index and reduce the anti-nutritional factors below 3.04 milligrams. And for this, we have optimized 92 to 96 soya prototypes by giving them different time, duration, pressure, and heat treatment in this processing technology. And the second one is the evaluate protein quality. Our hypothesis is that this EPS improves the PDI value, trypsin inhibitor level decreased, and cure solubility increased in all soya prototypes, which are produced as a result of this hydrobarothermal technology. And the third hypothesis is regarding amino acid digestibility. As you know that if we are able to achieve this or we are able to protect the protein quality of soybean, so in this way we can increase the amino acid digestibility in the birds. And measuring at the end, our hypothesis is that this hydrobarothermal technology also improves the feed conversion ratio, feed intake, and egg mass in the birds. So for this, we have analyzed, first of all, at the lab stage, we have developed 92, 96 different soil prototypes while giving them different time, temperature, pressure, and moisture level at different parameters. And the second stage is the in vivo digestibility trials at farm level. In this trial, we have used 972 laying hens, which includes 4 replicates per treatment, 3 birds, and we have evaluated the growth performance, feed efficiency, and nutrient absorption. And here in this slide, I have enlisted these are our target parameters that we want to achieve this in this processing technology, like temperature, pressure, and moisture level. And these are our target milestone because total protein is more than 38%. is our target at first stage, and trypsin inhibitor level, it must be below than 4 milligram per gram. PDI value, moisture, and for it illness must contain 12 to 13%. And here is the second stage in the farm level. These are the parameters which we have evaluated at farm level. We have analyzed apparent and standardized ileal digestibility of amino acids, fat digestibility, live weight, feed intake, and feed conversion ratio. We also analyzed the egg quality parameters and intestinal viscosity. And here are the key results of our preliminary experiment at farm level about regarding these 92 soya prototypes. We are able to achieve this target, which we have mentioned that we want to reduce this trypsin inhibitor level below 4 milligram, and our target value we have achieved is 3.04. And the PDI value must be more than 20%, and we achieved 24%. And the protein digestibility index, 36 out of 80 soya prototypes give these results, which shows more than 90% protein digestibility index in poultry. And these are our milestones which we have met in our preliminary experiment. Out of 80 soya prototypes, only 36 prototypes shows more than 90 protein digestibility, and only 30 soya prototypes out of 80 prototypes shows more than 3,300 kilocalories apparent metabolizable energy, and only 4 prototypes shows more than 97% fat digestibility index. And trypsin inhibitor, we have achieved 3.2% in best of the best soya prototypes. And here's the comparison between the control group and our experimental soya prototypes at farm level. Feed intake, we have achieved the significant values in terms of feed conversion ratio, feed eggs quality parameters, and most important parameter is the ileal protein digestibility. We have achieved more than 92.1% in this technology. And at the conclusion, this hydrobarothermal processing technology reduced this trypsin inhibitor value from 4 to 3.04 and increased the protein digestibility index more than 24%, and protein digestibility exceed more than 90%, and this amino acid apparent metabolizable energy more than 3,300 kilocalories energy, which is our target parameters, and we successfully achieved in our preliminary experiment. And the third conclusion is that we are able to improve the feed conversion ratio, egg quality parameters in our hypothesized EPS soya. And at the end, as you know, that if the protein is more than or above 90% digestible for the bird, so in this way we are able to achieve the European Union sustainability goals. Because in this way we are able to increase the digestibility and overall reduce the nitrogen and phosphorus excretion in the environment. And here is a take-home message from our today's presentation, that this hydrobarothermal processing technology, locally produced from non-GMO soybean, high quality parameters it contains. As you know that we are able to achieve this while giving different time, temperature, and pressure level. And at farm level, we are able to achieve this target while using 972 adult laying hens. And in the third step, EU sustainability goals, because as I mentioned that we are working on the sustainability goals as well. If the protein is more digestible, so in this way we are able to achieve this target. So thank you so much for your attention. If you have any question, I would be happy to take it. Thanks so much.
Thank you very much, Muhammad Umair, for a very nice presentation. I know you had a very fantastic set of data and you rushed it through because of the shortage of time, but we can see that you have done a fantastic work there. Because of the shortage of time, I think we have to skip the questions. If anyone has any question, please write it in the question chat box, and Umair would be more than happy to answer it afterwards. With this, I think it brings us to the close of session 1. Please join me to sincerely thank all of our keynote speakers, oral presenters, and participants for their outstanding scientific session. Today's presentation has taken us on a remarkable journey across the poultry production, whether it was via embryonic development, skeletal development or skeletal health, to gut programming, feed innovation, and circular nutrition schemes. What we have learned, or what is emerged from this session, is that the nutrition remains one of the most powerful tools available for improving poultry health, welfare, and productivity. The next theme that emerged from this session was that sustainability is no longer a future aspiration, but a present-day necessity requiring innovative approaches to feed resources and resource utilization. The third scientific progress is accelerated through collaboration, and today's session has demonstrated the value of bringing together expertise from different countries, institutions, and disciplines. I would also like to thank our graduate student and early career researchers for their fantastic work and presentations. I hope today's discussions have inspired new ideas, new questions, and perhaps even future collaborations. I would like to thank all the participants for their engagement and contributions. I wish everyone an enjoyable and productive remainder of the session, and would really like to thank the PPP and GRC for organizing such a fantastic conference. And at the end, I would like to say a big thank you to everyone, and Allah Hafiz.
Before concluding this session, I would like to express our sincere appreciation to our session chair, first scientific session chair, Professor Dr. Frina Khattak, for serving as session chair and for guiding today's scientific discussions with the exceptional leadership and expertise. So we are very much thankful to Professor Dr. Frina Khattak for your valuable contribution to the GRC 2026. So we now have a very short certificate distribution to our distinguished keynote speakers as well as to our oral presenters. So for this, I would like to ask Professor Dr. Gibran Hussain and Dr. Nasir Mukhtar to join us along with Professor Dr. Frina Khattak to present the certificates of presentations to our oral presenters, as well as the certificates of appreciation to our keynote speakers as a token of love. So, Professor Dr. Frina Khattak, it's over to you.
Okay, the first appreciation certificate is for Professor Kim. Professor Kim, we are really grateful to you for sparing your time and sharing your valuable presentation with us. Thank you once again for your time and presentation. I don't think we have Professor Kim with us. Okay.
Now we have Professor Dr. Eugenie Rora from the Queensland University of Australia.
Yeah, Professor Rora, we are really grateful. We would like to thank you for your time, your presentation, and for your very innovative talk on Innovo technique. We are really grateful for your presentation, and this certificate is for your appreciation. Once again, thank you so much.
Thank you so much, and congratulations to the organizers. It's been fantastic. You've had a wonderful success, and thanks everyone for listening and for inviting me. It's been a pleasure. Thank you.
Thank you so much. Thank you once again.
Thank you, Professor Rora.
And we would also like to thank Professor Sumas for her presentation. This is the certificate of appreciation for your time and your presentation. Once again, thank you so much, Professor Sumas.
So now this certificate goes to Professor Dr. Frina Khattak for as a keynote speaker and for chairing the scientific session. We are very grateful for having Professor Dr. Frina Khattak. So This is
Thank you very
our
much.
token of appreciation to you.
Thank you. For oral presentation, I would really like to thank Dr. Abdul Ghayas for your presentation. And we appreciate that you took time and shared your research work with us. And we wish you all the best in your future career as well as your future research work. Thank you very much, Dr. Abdul Ghayas. With the concluding certificate, I would like to thank Mohammad Umair Asghar for a very fantastic presentation. And once again, we know the time was short, but you did a fantastic job of presenting and narrowing down your research in just 10 minutes. Thank you once again for a fantastic presentation. And this is the certificate for your certificate of appreciation for your fantastic presentation. Thank you so very much. And I think this concludes this session.
So now, yeah. Thank you very much, Professor
You're
Dr.
welcome. Thank you very much.
We
Thank
now
you.
have a 10-minute short break to have some coffee or tea. Please enjoy your coffee or tea. So we will start with at 12:30 p.m. Pakistan time. We will start with the scientific session number 2 on poultry diseases and gut health management. And this scientific session will be chaired by Professor Dr. Muhammad Kashif Salimi, Chairman, Department of Pathology, University of Agriculture, Faisalabad, Pakistan.
Okay, thank you very much.
Everyone.
Bye. Allah Hafiz.
Allah Hafiz, ma'am. Thank you so much. Ladies and gentlemen, a few more minutes as we will be joined by keynote speakers from the United States and from Canada, and it's 2:30 a.m. right now there. So please, thank you very much for your patience. We will be starting the session very soon. Okay, ladies and gentlemen, and thank you very much for your kind patience. We now move to our scientific session number 2 on poultry diseases and gut health management. This session is chaired by Professor Dr. Muhammad Kashif Salimi, Chairman, Department of Poultry Production at University of Agriculture, Faisalabad, Pakistan. So now, without further delay, I would like to ask Professor Dr. Kashif Salimi to please give a very brief welcome remarks to this session. It's over to you, Professor Kashif.
Hello.
Yes, we can hear you.
Okay, thank you. Thank you, Sam. Welcome all of you to Research Conference 2026 that is being organized by the Poultry